Are we live now? I guess we are live. Okay, it's fixed, it's sorted. Hello everyone, welcome to PW NEET English YouTube channel. We're back. I mean, I kept talking the entire time thinking that I was at least visible and audible and it was at my end there was a glitch, but turns out you could not hear or see me. So hi, good evening everyone. How's everything going? How's your preparation going? Okay. Hi Shalini, Inchara, Hridyanjali. Good evening. Hey Henry, good evening. Okay, so on a scale of 1 to 5, where would you rate your preparation? 5 being like you're fully prepared. If I give you an exam right this moment, you'll be like, you'll just ace it. Hey Alice, good evening. Hridyanjali, I'm doing great, as fabulous can be. Thank you for asking. Okay, Inchara's rating a 4 out of 5. Cool, guys. So, in the last session of mine, we started with the 11th grade syllabus. We finished one 11th grade session where we discussed, I guess, 5 chapters. Today, we're starting with the 12th grade syllabus where we'll be discussing 3 chapters, 3 high-weighted chapters, which are sexual reproduction in flowering plants, principles of inheritance and variation, and molecular basis of inheritance. So we can revise all of these chapters very quickly in one shot. If you're ready, give me a thumbs up in the chat and we can get started. What happened? Mouse, it has stopped. Abhinaya is asking where you can find the class notes. Class notes will be available on the WhatsApp channel. I guess the WhatsApp channel's link is pinned in the chat and it's also available in the description. You can find it there. You will get it only after the class is over. Maybe after like 15-20 minutes after the class ends, you will get access to the notes in PDF form. All right. So now that everyone's ready, let's get started with sexual reproduction in flowering plants. What are flowering plants? We've already discussed plant kingdom. So you know that in the plant kingdom, there are 5 divisions, of which flowering plants are angiosperms. These are the only plants that produce flowers. Of course, we don't see flowers blooming on top of a Christmas tree or on a cypress tree or on a fern, right? It's only in the angiosperms that flowers are produced. So this chapter, as the name itself indicates, will talk about how sexual reproduction happens specifically in our angiosperms. Now, the entire process of sexual reproduction can be grouped into 3 categories. If you talk about sexual reproduction in any higher organism, be it an animal or a plant, it essentially involves formation of gametes and fusion of gametes, right? The male gamete forms, the female gametes also form, and the male and the female gametes fuse to form what is called the zygote. And this process is called the fertilization. So, of course, fertilization happens even in sexual reproduction in flowering plants. But here, the fertilization process is kind of unique compared to other plants and other organisms because 2 fusion events will take place, and that is called double fertilization. So, one of the major events of sexual reproduction is double fertilization. Of course, we learn about that. But before fertilization happens, gametes need to form, right? So, the structures responsible for producing gametes need to form. A lot of events need to take place. Whatever structures are essential for, sexual reproduction and fertilization to take place, all of that we learn about in pre-fertilization structures and events. For gametes to form and fuse, for gametes to fuse, they should first form, right? By what process exactly gametes form, where it forms, what is the structure of that, all of that we will learn under pre-fertilization structures and events. And after fertilization, what is the further development, what structures form, what processes happen, that we will learn under post-fertilization structures and events. I'm sure you know pre means before, post means after. right? So here's what happens before fertilization, here's what happens after fertilization. Now, when you consider a flowering plant, it's not like the moment the seed germinates, a flower will start blooming, right? Just like how in many animals, as soon as the baby or the young one is formed, it will not have the ability to reproduce. Consider humans for that, as an example. So when humans are formed, the moment they are formed or immediately after that, or if after a few days, they will not attain the ability to undergo sexual reproduction. Same goes for plants, okay? Initially, the plant has to grow vegetatively, photosynthesize, make sure the reserve food is stored in the plant. It grows considerably, produces more leaves and branches and all of that. Only after that period of time, after a period of vegetative growth, will flowering be seen in plants. Okay. So before a plant decides to produce a flower, it will have to, analyze the environment it is present around. I mean, that is present around it. Okay. So someone's asking if we'll be solving questions today. Not today. Once this whole theory series is over, maybe after the next 1 week or 1.5 weeks, we will start solving questions, only questions then. Right now we're doing only theory. After this series, we will be solving only questions. Okay? Yeah. So, before the plant produces flowers, it will take note of all the environmental factors around it. It will check for whether the temperature is good or not. It will check for whether the soil has sufficient nutrients or not. If it is a too dry a period, it will check for all of that. Is the sunlight sufficiently available for photosynthesis or not? Because producing a flower means producing the next generation. It is through the flower that seeds will form, right? So when seeds form, seeds are like the babies for the parent plant. So when the baby is forming for the plant, it should make sure that when the seed is formed and it germinates, the conditions should be suitable, right? So the leaves will measure the duration of the day length. It will measure what the temperature is and all of that, and will send signals to the shoot apex. Normally, what does the shoot apex do? It will start giving out leaf primordia, which eventually transform into leaves. When the conditions are favorable for sexual reproduction, if the conditions are just optimum for the plants, then it will send a signal, it will send certain hormonal signals from the leaf to the shoot tip. Once the shoot tip receives those hormonal signals, then it will start producing floral parts instead of leaves. Okay, so that is how flowers are formed. Instead of a leaf, a flower will arise from the shoot apical meristem. Okay, flowers or floral buds will arise from the shoot apical meristem. Okay, now let's talk about flowers. In angiosperms, flowers are considered to be the sexual reproductive part. When we call any organ a sexually reproducing organ or a sex organ, what we essentially mean is that within those organs, formation of gametes will take place, right? If you consider humans, we call ovaries in females the sex organ and the testes in males as the male sex organ. The reason the testes is called the male sex organ is because within the testes is where the sperms are produced, the male gamete is produced. Within the ovaries is where the female gamete is produced. That's why it's called the sex organ. Likewise, it is within certain parts of the flower that the male and the female spores are produced here. That is why it is called the sexual reproducing organ in flowers. Okay, so we've already discussed the structure of a flower and morphology of flowering plants. We'll do like a quick recap. This is what a typical flower will look like. Of course, there are so many different variations in flowers. Some flowers don't have petals at all. They have just one accessory whorl. Some flowers don't even have a stalk or the pedicel. They are sessile. Some flowers have only androecium. Some of them have only gynoecium. Of course, you have a lot of variation when it comes to flowers. But here's what generally a typical flower in an angiosperm will look like. It has a stalk that is called the pedicel. At the tip of the stalk, you have a broad base-like expanded region which we refer to as the thalamus. Okay, so here we have the thalamus. On top of the thalamus, in concentric circles, you have 4 different parts of the flower arranged. Outside to inside, this order will never change. We call these concentric circles as whorls. Starting from outside to inside, the outermost whorl is called calyx. Okay, so calyx is made up of these small green leaf-like structures that are called sepals. Collectively, it's called the calyx. Right inside the calyx, the second whorl from outside is made up of petals. Petals, they are usually brightly colored in order to attract pollinators. The entire whorl of petals is called corolla. Uh, who says Jagriti? Jagriti is asking if this is for droppers. Dropper, fresher, doesn't matter. This is for anybody who's aspiring uh, to become a doctor by giving the NEET 2026 exam. Doesn't matter if you're a dropper or not a dropper. This is a quick revision series where we're discussing or revising, in fact, many chapters in one shot. So yeah, हिंदी नहीं आता पावर प्ले वर्स चैनल का नाम देखो PW NEET English. Okay, all right. So yeah, I was saying that the petals are meant to attract pollinators. What do we have next? Right inside the whorl of petals, we have the male reproductive part, which is called the stamens. The entire whorl of stamens is what we refer to as the androecium. It is the male reproductive part of a flower. It is within the androecium that the male spores are produced. Innermost whorl in a flower is what we call the gynoecium. Gynoecium is also called the pistil. Don't get confused with the terms here. Gynoecium and pistil mean the same thing. Its units are called carpels. That is the female reproductive part. Since our focus here is not to study about how is the estivation, how is the sepals, does it have epicalyx or not, we are not worried about its morphology. We are here in this chapter to study about how sexual reproduction happens. So moving on, we will not care about how is the sepal, how is the petal. We will focus mostly on the reproductive parts, which is the androecium and the gynoecium. Okay. Hi Hakshita, good evening. Power play was aaj nahi hoga question practice. After this series, there will be another series where we will only do questions for like the entire day. So then we will be doing questions. Okay, not today. Cool. So we'll start off learning about the structure of androecium. If so far everything is clear, give me a thumbs up in the chat. We'll start off with the stamen. Stamen, like I mentioned earlier, is the male reproductive part of the flower. The entire whorl of stamens is what we refer to as the androecium. A stamen typically has two parts. It has a long, slender filament. This filament on one end proximally is attached to the thalamus of the flower. On the other end, it bears a knob-like structure which is called the anther. The anther, in most cases, in majority of the cases in angiosperms, of course, every rule has an exception, but in most cases, it has two lobes, which is why we call it bilobed. You can see how this is one lobe here and this is the other lobe here. Two lobes are there. The two lobes of the anther are connected by what is called connective. Okay, so in between these two lobes, there will be parenchymatous tissue that is keeping the two lobes of the anther together. So that's what bilobed means. If you look at one lobe here, within each lobe, you have two cavities or chambers of sorts, which we call theca. Since each lobe has two theca, we call it dithecous. These terms are important and you must remember what they mean. In between these two theca, present within the same lobe, you can see like a groove running longitudinally throughout the length of the anther. And the anther is a four-sided structure. It is tetragonal. Within the anther, you have four microsporangia present. Now, don't ask me what is microsporangia. Of course, you can ask me what is microsporangia. So, yes, Jagriti, it will definitely help you for revision, regardless of whether you're a fresher or a dropper. What we're doing here is revising. It should help anybody, even if you're like giving your board exams in 12th grade. It's the same chapter, the syllabus is the same. So, it'll be like a revision for you. Okay. Who is this? Alden. Hi, Alden. Enian. Enian, na? Okay. I don't know if I'm pronouncing your name correctly, but hi. Yeah. So within each lobe of the anther, here is one lobe of the anther. Within this lobe, you can find there are two microsporangia. Within the other lobe, there will be two microsporangia. What are microsporangia? Sporangia are structures within which spores are produced. Microsporangia are those types of sporangia within which microspores are produced. This is the male reproductive structure. You know that in angiosperms, the main plant body is a sporophyte. So we're talking about the sporophytic plant body here. What is the sporophyte supposed to do? It's going to produce spores. The male part of the reproductive structure will produce the male spores or the microspores, and the female part will produce the female spores or the megaspores. Right now, we're talking about the sporophyte producing the male microspores. That happens within the anther. Within each bilobed anther, that is dithecous, you can find four microsporangia. So that's why we call it tetrasporangiate. Tetra meaning 4. Because it has 4 sporangia, we call it tetrasporangiate. Okay, so remember, bilobed is because it has 2 lobes. Dithecous is because each lobe has 2 theca. Tetrasporangiate is because one anther entirely, both lobes included, has a total of 4 microsporangia. That's why you call it tetrasporangiate. Now, we have to understand the structure of the microsporangium here. A microsporangium, within a microsporangium, pollen grains, okay, you're in Yaqeen, Alden, hi. Maybe not very active in the chat, so that's why I don't remember the name. So, yeah, the microsporangium is a very, very essential and important structure for a plant because it is through the microsporangium that microspores get produced. If the microspores don't get produced, then the continuation of generation and the completion of life cycle will not happen. Within the microsporangia, microspores are produced. These microspores eventually go on to become the pollen grains. So, within the microsporangia, very precious, important cells are going to form in the future. So, it requires like full security, like VIP level security, it requires. So, the wall of these microsporangia are extremely thick walled, and within the wall of the microsporangia, you have 4 different types of layers. So, if you take a section of an anther, it will appear like this, like a butterfly shaped outline. This represents one lobe, this represents one lobe, and this is your other lobe. Like I mentioned earlier, the 2 lobes are held together by what is called the connective. So, this is the wall of the anther. The wall of the anther is made up of 4 different types of cell layers. They are meant to protect and nourish the developing microspores. Okay. So, if you look at the wall of the anther, let's focus on just one microsporangium for now. If you zoom into one microsporangium, here's what it looks like. I'll tell you what these different wall layers are. Like we saw in the chapter anatomy, the outermost layer of the stem, root or leaf was the epidermis. Likewise, even the outermost layer of the anther is called the epidermis. Epidermis is a single layer of cells that is obviously protective in function. Next, right underneath the epidermis, you have a layer of cells called the endothecium. The endothecium also offers protection. What is it protecting? Whatever is lying inside, we'll come to that. Within the endothecium, you have 1 to 3 layers of cells called the middle layers, and the innermost layer is the tapetum. This is in order of how it is present here. Outside to inside, here's how it is arranged. This sequence you should be sure of. This is outside, this is inside. Okay. Over here, the epidermis, endothecium, middle layers, they all, when the anther is young, will protect it. Okay, because inside the anther you have what is called microspore mother cells. These microspore mother cells are going to become microspores in the future. It needs protection. So, when the anther is young, these 3 layers will offer protection to the developing microspores. But when the anther matures, what do we mean when we say the anther has matured? It would have already formed the pollen grains. What's the point of developing pollen grains and protecting it inside like a thick wall? After the pollen grains are produced, they have to be released outside so that they can get pollinated and bring about fertilization. So once the anther is matured and the pollen grains are formed, the exact same wall layers that initially offered it protection are now going to help, disperse the anther. Okay, so that process is called dehiscence. So the same 3 layers will help in dehiscence of the anther. This innermost layer of the wall called the tapetum will provide nourishment. What will it nourish? It will nourish the microspore mother cell. See, this is the wall layers. Till the tapetum, it's the wall layer. Inside that, you have sporogenous tissue or microspore mother cells. These microspore mother cells are eventually going to divide and give rise to the pollen grains or microspores. So, these sporogenous tissue or the microspore mother cells require nourishment for the microspores to develop properly. That nourishment is provided by the innermost layer of the wall That's the closest, right, to the sporogenous tissue. They're literally neighboring cells. So this will nourish the developing microspores. That's its job. And what is the function of the microspore mother cells? As the name itself indicates, they are mothers of microspores, which means they will undergo division to give rise to microspores. So they are involved in the formation of microspores and pollen grains. Okay? Jagriti, you're determined to crack NEET in my first attempt. What should I do now? I'm currently appearing for board exams. If you're already if you're already part of the Yaqeen NEET 2026 batch, you know that the syllabus was completed long back. I hope that you're fully prepared for your board exam right now. If you're prepared for NEET, then board exam is like a walk in the park, right? So, of course, you will need a different kind of strategy and preparation for your board exam, but conceptually, you will be very sure of all the chapters, right? I'm assuming that. Okay. So what you can do after your board exams is start giving more and more mock tests, and also don't aim for, I'm not saying that you shouldn't aim for 720 or anything. If there are some weak chapters that you have, instead of, randomly going about with those chapters, you make a list of which are the high weighted chapters and approach them accordingly. But I want to know where you stand right now. Like, have you finished watching the lectures of all the chapters? Have you read NCERT? Have you taken our tests? Is there anything that you have not done? I'm assuming that since you were a part of the batch, you have done everything, and that's why I'm giving you this advice. But if there's any blocker or something, then you can let me know. If there's any issue or concern that you're specifically facing, you can tell me. At least for biology, I think I finished the syllabus and Vindhya ma'am finished her syllabus 2 months ago at least. So you should have completed your part of the preparation of those chapters by now. Since what you're asking is kind of vague, Jagriti, I can't give you like a specific answer. Okay? Yeah, so which college do you want to get into? What is the safe score in that particular college based on the last few years? What is your current score in the mock test? Analyze all of that. After giving your mock test, check for where you're making mistakes. Why are those mistakes happening? Do you not understand those concepts well? Go revise those concepts. After each mock test, you should get better. Okay, so that's the whole idea of taking mock tests. You should get better with time management. The speed and accuracy of reading questions should increase. So you work on all of that, and don't take too much stress. Okay. All right, Jagriti, I said so much. I don't even know if you're still here or you just left. Okay, we'll move on. Yeah. So I told you that the microspore mother cells present at the center of each microsporangium is going to give rise to microspores. We'll learn about how those microspores form. So the process by which microspores form is called microsporogenesis, and clearly it happens inside the anther. During microsporogenesis, what happens is the microspore mother cells that are present inside the microsporangia, them being neighboring cells, will obviously have cytoplasmic connections between themselves. So plasmodesmata will be there. The first thing that they do is they will cut off those plasmodesmata by synthesizing a thick callose wall around itself. So it's like each of these cells will get separated from each other, no communication. Then it's called then the microspore mother cell will just cut off communication with the neighboring cells by producing thick walls around itself. Then what it does is it will undergo meiotic cell division. Remember this, whenever we talk about gamete formation, you may have heard that during gamete formation meiosis happens, but that's not really true with respect to plants. We've already seen that in plant kingdom. In plants, what happens? When spores are forming, meiosis will happen. Sporophyte giving rise to the spores, meiosis will happen. Gametophyte producing gametes, mitosis will happen. During gamete formation in plants, mitosis will happen. During spore formation, meiosis will happen. Right now, what is forming? Spores or gametes? Right now, spores are forming. So, right now, meiosis will happen. The microspore mother cells, being part of the sporophytic plant body, will be deployed in nature. These diploid cells will undergo reductional cell division called meiosis, as a result of which 4 haploid cells will form. Initially, all of these 4 haploid cells will be present within the same callus wall. And since they are all 4 present together, you call that a microspore tetrad. Tetra means 4, right? 4 of them will be present within the same wall. Later, by some tapetal secretions, the outer wall of callus will disintegrate, break down, and these 4 cells will be separated. Once they are separated, you can call them pollen grains. They are haploid. Is it clear? So, this entire process by which the diploid microspore mother cell undergoes meiotic cell division to give rise to haploid microspores is called microsporogenesis. Okay? There is no revision in the batch. The revision is happening here, Alden. So, you can join these lectures. Alright, so now that the pollen grains are formed, let's understand a little bit about pollen grains. Pollen grains are the male gametophytes in angiosperms. They appear in so many different shapes, but most of them are spherical in shape. They're super tiny, they're very small. They measure about 25 to 50 microns in diameter. Okay, as you can see, they come in so many different shapes. So, a pollen grain is also a very important part of the plant, right? It's a very important cell because it is within the pollen grain that the male gamete will form. It is through the pollen grain that sexual reproduction takes place. So, pollen grains, once it is released, till it does its job, which is going and fertilizing the ovule of the flower, it should be stable, right? So, its wall is also very, very thick. The wall of the pollen grain has 2 layers. The outer wall is called exine. It is made up of a substance called sporopollenin. And this sporopollenin is one of the toughest substances that you can find in the living world. So, this sporopollenin cannot be degraded by any enzymes. It cannot be broken down by very high temperature, strong acids or alkali, which is the reason why pollen grains are preserved very well as fossils. Now, this is the outer layer. The outer layer is not continuous. As you can see, this orange layer is not continuous. There are certain regions where there is no sporopollenin in the exine. So, those regions where sporopollenin is absent, such regions are called germ pores. What are they called? They are called germ pores. It is through the germ pores that pollen tube will form. Inside the exine, you have what is called the intine. Intine is a continuous layer, no pores here, and it is made up of like regular plant cell wall material, cellulose and pectin. The microspore, when it formed, will have only one haploid nucleus within itself. That single haploid nucleus will undergo mitotic cell division to give rise to 2 cells. Now, when the microspore undergoes cell division, it will not undergo equal cell division, it will undergo unequal mitosis such that one large cell and one small cell is formed. The large cell is called the vegetative cell and the smaller cell is called the generative cell. They are both haploid because they formed from haploid microspore, right? So, the vegetative cell is large, it has vacuoles typical of plant cell and it will have a large irregular nucleus. It will have abundant reserve food stored in it. The smaller generative cell is spindle shaped. It will float in the cytoplasm of the vegetative cell. Okay, so this will also have a prominent nucleus, but compared to the size of the vegetative cell, the generative cell is much smaller. Okay, so this kind of floats in the surface, not in the surface, in the cytoplasm of the vegetative cell. I told you that pollen grains are the male gametophytes, but so long we haven't yet found the gametes formed, right? So, gamete is not yet formed. I named 2 cells, the vegetative cell and the generative cell, but gametes are nowhere in sight yet. So, unless it forms a gamete, we cannot call it a mature gametophyte. What happens next is, this generative cell, the Kutti cell that is present here, this will undergo mitotic cell division. Once it undergoes mitotic cell division, it will give rise to 2 male gametes. Okay, so this cell will divide by mitosis and produce two male gametes. So a pollen grain that contains one vegetative cell along with two male gametes is called the three-celled stage of pollen grain or the mature male gametophyte, because right here the gametes are formed, so it's safe to call it a gametophyte. Now here you have the two-celled gametophyte. This is three-celled, this is two-celled gametophyte. In majority of angiosperms, pollen grains are released in the two-celled stage. Like more than 60% of angiosperms, pollen grains, when they liberated, they are in the two-cell stage. The second mitotic division will undergo after the pollen are released, and in the remaining around 40% of pollen grains, it is released in the three-cell stage. Okay? Okay. Meiosis happens only in the spore formation, right? Yes, meiosis happens during spore formation for the formation of spore, because sporophytic tissue is diploid, spores are haploid, meiosis has to happen. When gametophytic, when gametes are forming in the gametophyte, as you saw in this case, this is the male gametophyte. In this gametes are forming, so during gamete formation, mitosis itself will take place. Okay? Alden, Jagriti, I'll answer your question shortly. We'll finish off one concept and then I'll take up your questions one by one. Okay? Alright. So that's about the pollen grain structure, its two-cell stage and the three-cell stage. Now we'll talk about some disadvantages that humans have because of pollen grains and some advantages. You may have noticed how a lot of people develop allergies because of pollen, especially because of Parthenium plants. Parthenium plant is also called carrot grass. It is not native to India. It was, introduced into India as a contaminant. It came to India along with imported wheat, and it has never left ever since. So the pollen grains, because they're very tiny and dust-like, they can cause a lot of issues in the respiratory tract of sensitive people. So pollen grains of many species such as Parthenium are known to cause pollen allergy. So many disorders such as asthma and bronchitis can also arise in individuals because of inhaling these dust-like pollen grains. Pollen grains' advantage is that it is rich in nutrients. That is what bees and insects eat as food, right? So it is of course rich in nutrients. Because it's rich in nutrients, it's consumed by humans. It's available in the market in the form of tablets and syrups. So apparently it's also used to enhance the performance in athletes and race horses too. So that's an advantage of pollen grains that for our lives. Okay. Yeah. So there's something called pollen viability about which we should understand. Pollen viability. See, what is the pollen grain's job? The pollen inside is carrying gametes, right? There are two male gametes present inside the pollen grain. The pollen grain has to go land on the stigma. Once it lands on the stigma, it has to form a pollen tube and release those gametes inside the female gamete so that fertilization happens. It's not like the pollen grain is attached to the parent plant forever, right? The parent plant, after producing the pollen grains, will release it. So once it is released, it will not have unlimited supply of nutrients. For as long as the nutrients stored or conserved or present in the vegetative cell is available, they can somehow manage to survive. But once all of that is exhausted, they cannot survive any longer because they're still not attached to the parent plant. They do not get those nutrients from the parent plant. Once they are released, they are to themselves, right? So it's not like the pollen grains will carry or retain the ability to bring about fertilization forever. Every species of plant will have its own pollen viability. In some plants, like in some grasses like wheat and rice and all of that, once the pollen grain is released, it will have the ability to fall in a stigma and bring about fertilization only within 30 minutes. Beyond that, it will lose the viability. So if a pollen grain is released from a wheat plant and it lands on a stigma after 45 minutes, it will be futile, it will be useless because it would have lost the ability to form a pollen tube and bring about fertilization. So the duration uh, which a pollen retains the ability to bring about fertilization, that is called pollen viability, and that varies from one species to another. Okay, so once they are shed, pollen grains have to land on the stigma before they lose their viability to bring about fertilization. The viable period is variable. It also depends on environmental factors like temperature and humidity. In some cereals, grasses such as rice and wheat, within 30 minutes of their release, they should bring about pollination. Otherwise, they will lose their viability. But in some families, like in Rosaceae, Leguminosae and Solanaceae, it will retain this ability for a few months. Pollen banks are places where pollen grain is stored. Okay, see, for crop improvement programs, let's say you're a farmer or you're a plant breeder and you have two different varieties of rice. One is like super long basmati kind of rice and another one is a pest resistant variety of rice. You want to make a hybrid between these two varieties. You want to take the pollen grain and dust it on the you want to collect the pollen grain, dust it on the stigma, and you have to do all of this, right? If you have to do all of this and within 3 minutes, if the pollen loses its viability, it's difficult for us to perform these hybridization sort of experiments. And even if you want to store pollen for long-term use for any breeding programs, if it has very little viability, you can't store it very well. So that's why they store these pollen grains in pollen banks where their viability periods are kind of extended. So can be kind of extended. So such pollen banks store pollen grains at very low temperatures as low as minus 196 degrees Celsius, and it is stored in liquid nitrogen, and that's called cryopreservation. So whoever is saying Sumit, cylinder nahi mil raha, oh my god, do you think like this is a cylinder shop and you putting like crying emojis here is going to help? Where do these people come from? कौन है ये लोग? Just give me 1 minute. Okay. इलेक्ट्रिक गैस नहीं हेनरी, इलेक्ट्रिक स्टोव होता है. ऑल राइट. Okay, so we'll move on, guys. That's about pollen viability. Now that we're done with the hero, we'll start off with the heroine, which is the gynoecium, female reproductive part of a flower. We've also learnt about the parts of the gynoecium and its morphology in another chapter called Morphology of Flowering Plants. For right now, our focus is going to be on how is gynoecium involved in sexual reproduction. So we look at that. So gynoecium is also called the pistil. Its individual units are called carpels. The number of carpels can vary from one species to another. It can be one in some plants and it is many in some plants. If it's a single carpel present in the gynoecium, you call it monocarpellary, mono meaning single, obviously. If it has many carpels present in the gynoecium, then you call it multicarpellary. When there is more than one carpel present in the gynoecium, it can either be fused or it can be free. If it is free, then you call it apocarpus. If it is fused, then you call it syncarpus. Apocarpus kind of ovary can be found, or gynoecium can be found in Michaelia champaka, and syncarpus kind of gynoecium can be found in papaver. These are examples that are important. Not just these examples, every example that you come across in NCERT is important because questions can arise from anywhere. It can be asked from anywhere. So make sure that you've covered NCERT thoroughly and you've memorized all of the examples. Okay. Now, speaking of gynoecium, it has three different parts: a stigma, a style, and ovary. Stigma is the topmost part of the gynoecium. That is the landing platform for pollen grains. Pollen grains, if it comes in directly, falls near the ovary, even though it is the closest to the ovule. Pollen tube formation will not happen here. It happens only if it falls here. So this is like the official entry site for pollen grains inside the gynoecium. So this acts like the landing platform. It's such a cool thing that pollen grains and the stigma talk to each other by means of chemicals, and they're like, oh, see, I came from this plant. Do you like me? And it's like, no, 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 you like my brother. And it'll browse on it and it'll be like, no, no, no. And then it'll look for another pollen grain which has a different genotype. We'll talk all about that later. But stigma is pretty cool. Okay. So this stigma is what will ensure that, the right kind of pollen will come and form a pollen tube. And then you have a long, slender style. The style is kind of, I don't know, I wouldn't say useless, but yeah, it's what makes sure that the stigma is present, raised above, so that the pollen grains can come and fall on it. Otherwise, it does not have too much of an important function. And the lowermost part of the gynoecium is called the ovary, the bulged portion, that is called the ovary. Inside the ovary, you have a locule, a chamber will be present. Within that, you have a cushiony tissue called placenta present. And to this placenta, one to many ovules will be attached. How ovules are arranged inside the ovary, we've learnt about that in placentation, right? So stigma is the sticky region on top of the pistil. It serves as a landing platform for pollen grains. Style is the elongated, slender part that connects the stigma to the ovary. Ovary is the basal bulged part of the pistil, and it's made up of ovules. Like I mentioned, the cavity inside the ovary is called the locule. Within the locule, placenta is present. To the placenta, ovules are attached. The number of ovules varies from species to species. In some, it's only one. In some, there's many. In wheat, paddy, mango, you have only one ovule per ovary. Whereas in papaya, watermelon, orchids, pomegranates, you have many, many ovules inside the ovary. Okay. Uh, okay, Jagriti. I'll answer your question shortly. All right. So let's talk about the structure of one ovule or megasporangium. Similar to how within the anther you had microsporangium, within the ovary, within the gynoecium, you will have megasporangium. And these ovules are nothing but megasporangia. It is within these ovules that megaspores will be produced. So this is Megasporangium. Okay. We have to learn about the structure of Megasporangium. Megasporangium is a very important structure for the plant. Within that, the megaspore will form. It will ultimately transform into the seed, right? So even whatever megaspores are formed inside the megasporangium, they require high level security. So here also you have protective outer layers and all of that. We'll learn about its structure. So there will be stalks for ovules. The stalk is called the hilum. Okay. This is an inverted ovule called the anatropous ovule, where the body of the ovule fuses with the stalk or the hilum. Did I say hilum? I should have said funicle. I'm so sorry. My bad. The stalk of the ovule is called the funicle, not the hilum. I'm sorry. The point of attachment or the region where the body of the ovule is fused with the stalk or the funicle is what we call the hilum. So here you can see what I'm highlighting here is the body of the ovule. Okay. So this outer part is the stalk. You can see how the stalk is fused with the body of the ovule in this region. That point is what we call the hilum. That region where the body of the ovule fuses with the stalk is what we call the hilum. So within the ovule, you have two protective layers that are called integuments. No fancy names here. Outer layer is called the outer integument, inner layer is called the inner integument. Here you do not want these integuments to fully cover the inside of the ovule, because if you give it like that level of security, then the pollen tube will not be able to enter inside the ovule. So that much security is not required. You give security, but you give a little bit of freedom also. So these two integuments do not fully cover the ovule. There is a small pore or a slit sort of thing that is left behind for the pollen tube to form, and that is called the micropyle. You can see how it is not fully covered, right? There is a small opening present here that is called the micropyle. What is this essentially protecting? The two integuments of an ovule are protecting what is lying inside, which is the nucellus. It is the cells of the nucellus that will give rise to the megaspore in future. So this is what is being protected. Okay, this will form megaspore after undergoing meiosis. So outer integument, inner integument, inside you have the nucellus. Within the nucellus, the spore will form, the spore will give rise to the gametophyte and all of that will happen. So embryo sac formation also happens inside the ovule itself. The region of the ovule towards the micropyle is called the micropylar pole. And the region of the ovule away from the micropyle is called the chalazal pole. Okay, or the chalazal end you can call it. Now, let's understand how the cells of the nucellus will give rise to the megaspore. First, you have many cells in the nucellus. It's like parenchymatous tissue. All the cells are thin-walled and all of that. They'll appear similar, homogeneous initially, but once megasporogenesis is initiated, megaspore formation is initiated, one of the cells of the nucellus will start appearing different from the others. It will get differentiated, it will appear larger with a prominent nucleus and dense cytoplasm. And that cell is called the megaspore mother cell. Earlier during microsporogenesis, I told you how every cell present in the sporogenous tissue will end up undergoing meiosis to give rise to microspores, but here that's not the case. Not every cell in the nucellus will give rise to a megaspore. Within one ovule, one megaspore only will form. So for that, one cell only needs to divide. Not all of the cells here within the nucellus will undergo meiosis. So the one cell that will undergo meiosis will first start appearing like a diva. It will start looking very different from the other cells in its surroundings. It will appear bigger and have a prominent nucleus and stuff, and that is called the megaspore mother cell. This one, this central cell. See, initially all of them were of similar size. Now this lady has become much bigger, right? So this is the megaspore mother cell. This megaspore mother cell will undergo meiotic cell division. Once it undergoes meiotic cell division, obviously from one diploid cell you get 4 haploid cells. Even over here we get a tetrad of megaspores, 4 cells that are linearly arranged. But of them, 3 will degenerate, only 1 will remain functional. Okay. So from this, at the end of meiosis, you get 4 megaspores, that is called megaspore tetrad. Of them, 3 will degenerate and 1 will remain functional. These are gone and this will survive. So this is called the functional megaspore. What is a spore supposed to do? Spore will produce the gametophyte by undergoing mitosis, and this megaspore will do exactly that. This functional megaspore will undergo mitosis to produce the female gametophyte. Remember when we spoke about seed habit, I told you how for in angiosperms and gymnosperms both, female gametophytes do not have independent existence. The spore is formed within the sporophyte. Megaspore does not get released outside. Megaspore develops into the gametophyte within the sporophytic plant body itself. So this spore developing into the gametophyte happens within the ovule itself. Okay. So here you can see how this one single megaspore that is haploid will start undergoing cell divisions. What kind of cell divisions? Who's going to tell me? Is it mitosis or meiosis? It's going to undergo mitotic cell division. Miss Kim. Hi Kim. Hi Sohan. 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 So it's going to go undergo mitotic cell division. See, during mitosis, you have two phases. You have karyokinesis, cytokinesis. Karyokinesis is the division of the nucleus. Cytokinesis is the division of the cytoplasm. Here what happens is, karyokinesis happens very well, beautifully. The nucleus divides and gives rise to two nuclei. But immediately after karyokinesis, cytokinesis will not take place. So free nuclear divisions will keep happening 3 cycles. Okay. So first we started off with one cell having one haploid nucleus. That nucleus will divide to give rise to 2 haploid nuclei, which is called the 2 nucleate stage. Then again, these 2 nuclei will undergo another round of mitotic division and give rise to 4 nuclei. 4 nuclei will undergo another mitotic division, 8 nuclei. So initially the divisions are free nuclear, cytoplasm is not dividing, cell wall formation is not happening. So here you can see this is the 8 nucleate stage. Once there are 8 nuclei present inside the embryo sac, eventually these nuclei will reorganize and the cell wall formation will begin. Here you can notice how there are 4 nuclei at each pole. One of the nuclei from each pole will migrate towards the center, okay, and they will form 2 polar nuclei. They are called polar nuclei because they migrated from the poles towards the center. Now that these 2 nuclei from the poles have migrated towards the center, how many nuclei will we have at each pole? You will have 3 nuclei at each pole. Once that migration is over, cell wall formation will happen, and at the end of mega gametophyte formation, you will notice how at the chalazal end you will have 3 cells, at the micropylar end you will have 3 cells, and in the large, there will be a large central cell with 2 polar nuclei. See, in between these 2 nuclei, there will be no wall formation. They are both present within the same cell. So here you can see towards the chalazal end, you will have 3 antipodals, all of them are haploid. Towards the micropylar end, you will have what is called the egg apparatus. The egg apparatus includes 1 egg cell that is haploid and 2 synergids that are also haploid. Collectively, you call this the egg apparatus. Always remember that the egg apparatus is towards the micropylar end, antipodals are towards the chalazal end, and at the center you have a large central cell with 2 polar nuclei. They are both lying in the cytoplasm of the same cell, so there is no wall formation between these two. So you have a total of 1, 2, 3, 4, 5, 6, 7 cells. 1, 2, 3, 4, 5, 6, 7, 8 nuclei. So this female gametophyte is called the mature female gametophyte or the embryo sac. This has 7 cells and 8 nuclei. So it's called 7-celled 8 nucleate. 7 cells, 8 nucleate, 7-celled 8 nucleate stage. Okay. Oh my god, I fumbled so much there. Hi Manju. Good evening. Is it clear so far? Give me a thumbs up. Yeah. So with that, we finished the what part? Microsporogenesis, Megasporogenesis, formation of male gametophyte, formation of female gametophyte. Right now, male gametophyte is ready to bring about fertilization inside the pollen grain. Female gametophyte has the egg cell waiting for the pollen grain. We have to make sure that the male gamete meets the female gamete, brings about fertilization and makes a baby, and a seed will form and the next generation will arise, right? That's what needs to happen. So the pollen grain has to be released from the anther and it should go land on the stigma, right? So that's the next step. What do we call that? That process is called pollination. So the transfer of pollen grains, where were the pollen grains formed? They were formed inside the anther. The anther has to break open, dehyze, release the pollen grains outside to the environment, and the pollen grains have to go land on the stigma and bring about fertilization. This process by which pollen grains are transferred from the anther to the stigma is what we call pollination. Pollination can be of 3 types based on the source of the pollen. It can be autogamy, geitonogamy or xenogamy. Autogamy, auto means self. So autogamy is self-pollination. In autogamy, the pollen grain, okay. In autogamy, one plant is involved, one flower is involved. So one flower of the same plant is involved. So the anther of this flower is going to go land on the stigma of the same flower. This is called autogamy. For this to happen, the flower 100% has to be bisexual. Unless it is bisexual, self-pollination cannot happen. Also, secondly, there should be synchrony between release of pollen and stigma receptivity. So if that is not synchronized, then also it is not likely to happen. So when that is taken care of, if there's a bisexual flower where the pollen grains release and the stigma becoming mature are synchronized, are happening simultaneously, then self-pollination can happen. There are many plants in which you find 2 different types of flowers, chasmogamous flowers and cleistogamous flowers. The examples of these plants include oxalis, camelina and viola. Especially in Camelina, you have two different types of flowers, one produced above the ground, one produced below the ground. Who would have thought that flowers can be produced below the ground? Yeah. So let me enlighten you. Sometimes it's formed below the ground. And these flowers that are formed underneath, like underground, they are called cleistogamous. They are flowers that don't bloom at all. They don't open and expose their stamen and stigma at all. They are always in the bud condition. In such plants, self-pollination is only about to happen, bound to happen, because they don't open their petals and expose the stigma and stamen. So it's not likely for any other pollen to come and get deposited there, or the pollen from that plant to escape that flower and go elsewhere. So in such plants, invariably autogamy only will take place. There is no other way they can undergo any other type of pollination. However, in the same plant, you will have flowers that have exposed anthers and stigma. Such flowers are called chasmogamous flowers. Over there, self-pollination also can happen. Cross-pollination also is not entirely avoided. Cross-pollination also could happen. One advantage of self-pollination is, if at all the chasmogamous flowers do not receive pollen grains because no pollinators visited that flower, let's say, seed formation will not happen. But since these cleistogamous flowers are there, even if the pollinator does not visit the flower, they don't even need pollinators. Even in the absence of pollinators, fertilization will still happen. If fertilization happens, seed formation will 100% happen. So there you have assured seed set. Okay, so that is one advantage of these cleistogamous flowers. But continuously, if self-pollination keeps happening, that is also not necessarily a great thing for the plant. We'll talk more about that as we move on. Camelina, viola and oxalis are the examples for flowers that are cleistogamous, that don't bloom or open and expose their anthers and stigma. Moving on to geitonogamy. In geitonogamy, you have one plant involved but two different flowers of the same plant. The anther produces the pollen grain. This pollen grain will land on another flower but belonging to the same plant. This is called geitonogamy. This can happen in unisexual flowers or bisexual flowers. It is not essential for the flowers to be bisexual in this case. So this is very similar to autogamy, because though they are two different flowers, they are still part of the same plant, so they will have the same genotype. So genetically different pollen are not brought to the stigma. So functionally it is cross-pollination because it's happening between two different flowers, but genetically it is as good as self-pollination. Next you have xenogamy. This is like proper cross-pollination where two flowers belonging to two different plants are involved. Okay, so the pollen produced by the anther of one flower belonging to one plant, if it lands on the stigma of another flower belonging to a different plant, you call it cross-pollination. This is genetically going to bring newer type of pollen grains to the stigma because it will belong to a different plant, right? All right. So Manju is asking chasmogamous example. Chasmogamous, you give me the example, Manju. They are open flowers. Where can you find open flowers? Open meaning the uh, stigma and the anther is exposed. The flowers will bloom, the petals will bloom, open up. It will not forever be like a bud. Can you give me examples for that? Next. Agents of cross-pollination. For pollen grains to be transferred from the anther to the stigma, external agencies are required because pollen grains do not have locomotory structures. They can't walk on the road and go say, "Hi stigma, can I sit on you?" So they will have to be carried by some agent or the other. So here what happens is uh, living or non-living agents can help in the transfer of pollen grains from the anther of the stigma, from the anther to the stigma. If they are living organisms, then you call them biotic agents. If they are non-living abiotic factors, then you call it obviously abiotic agents. Biotic I'm sorry, excuse me. Yeah. So these biotic agents include living organisms like insects, certain birds, certain animals, certain other animals. Okay. Yeah. So that is what happens. Uh, so they are called abiotic agents. Among abiotic agents, what is commonly used for pollination is wind and water. Among wind and water, wind is more common. But the more common type of pollination is biotic pollination, where living organisms are involved. So let's see about wind pollination, water pollination, and pollination by living organisms or animals. First, let's talk about wind pollination. Wind pollination is called anemophily. Just give me a minute, I'll be back. I need. Okay. I'm running a cold and I have a little bit of a sore throat, but I'm okay. We can manage, no problem. Henry, thanks for asking. Okay, we'll continue. Yeah. So in the wind pollinated flowers, I'm chewing on these lozenges, like what do you call them? Like the coughlette sort of things. Don't assume I'm chewing pan or something. Okay. Yeah. So in wind pollinated flowers, you will find certain characteristics. We'll have to understand what are some of the characteristics of wind pollinated flowers. If they are pollinated by something like wind, which is not living, there's no need for the flower to be very fancy. The flower need not be colored. The flowers need not be very fragrant because wind cannot sense either fragrance or cannot see color, cannot go and eat the feed on the nectar from the flower. All of these are useless. So, in order to maximize pollination by wind, the flowers would have made certain modifications. Okay. So, what are some of those characteristics that you can find in wind pollinated flowers? Firstly, the flowers are not individual, large, and showy. Many tiny flowers are arranged in the form of an inflorescence. If you remember when we discussed the family Poaceae, I told you how the uh, inflorescence is there. I told you it's a spike of spikelets. The flowers are very tiny. We call them florets. They don't have brightly colored petals. They have very scaly perianth tepals that are called laudicules. So we saw, and I'm sure you would have seen the inflorescence of maize or ragi or uh, rice or wheat or whatever. So these grasses, very fancy flowers are not produced, but many tiny flowers are arranged in the form of an inflorescence. The pollen grains in any case are produced in large quantities because there's no guarantee that the pollen will go and land on the stigma. To increase the chances of successful pollination, the number of pollen grains will be very high in number. And these pollen grains that are produced by wind pollinated flowers will be very small in size and they will be non-sticky. If they are sticky, they will stick to random surfaces and successful wind pollination cannot be brought about. So they are very small in size and non-sticky. And yeah, because of their small size and less weight, they are not very dense. They do not fall off wherever they are released. Okay. So they are produced in large amounts. They are light and non-sticky. The stamens here are very well exposed. Well exposed means they come out of the tepals or the perianth members so that they can be easily carried by the wind, so that the pollen grains can easily carry the wind. The stamens here are very well exposed. What about the stigma? Stigma is supposed to trap tiny dust-like pollen grains. So the stigma is feathery here. Because it is feathery, it will make sure to trap the pollen grains very well. Okay. Each ovary will have only one ovule. This is most commonly seen in grasses. For example, in the corn cob, the hair-like structures that you find, that is actually the stigma and style of the flower. If you've seen in corn, like when you peel the outer green layers of a corn, you find these silky, nice blonde hair-like hair, right? So they're nothing but the stigma and style of these flowers. Okay. Now, let's talk about water pollination. Hi Swastika, good evening. Hi Manisha. Akshita. When will we post recordings of 11th grade botany? It will start from next week. In the next 3 weeks, one lecture each week. Botany syllabus of 11th grade will be completed, I suppose. Okay. All right. So we'll move on. Water pollination is called hydrophily. It's very, very rare when it comes to angiosperms. It's mostly seen in monocots. And there are just about 30 genera where you can find water pollination. Just because a plant just because a plant is aquatic, it is found in the water, it grows in the water, does not mean it will undergo pollination by water. For it to undergo pollination by water, it should get released in water. Water has to carry it to the stigma. Then you can say water pollination has taken place. But there are many aquatic plants like water lily and lotus in which flowers are not present inside the water body. Flowers are not submerged. Flowers are produced on the surface, right? If the flowers are produced on the surface of the water body, either wind or insects will bring about pollination, but not water. So only in plants that are submerged and produces flowers also within the water body in the submerged condition, only there can you find water pollination. You can find that in Vallisneria and Hydrilla, which are freshwater species, and Zostera, which is also called seagrass, which is marine. Okay. Ultimate KCAT crash course will start on the 17th. I think it's starting from 17th. Okay. So here, how does water pollination happen? We are talking about Vallisneria. It is an aquatic plant that is submerged, found within freshwater bodies. So here you have a separate male plant and a separate female plant. Male plant produces male flowers. Female plant produces female flowers. The male plant produces male flowers and releases that into the water body. So the male flowers of the pollen grains end up floating on the surface of the water body. The female flower is produced inside the water body. If this is the water body, okay, let's say this is the water body. The female flower is produced inside. But once the female flower matures, the flower, by means of its coiled stalk, initially it will be coiled inside and the flower will be here. But once it matures, it will uncoil the stalk and reach the surface of the water body. The male flower will swim on the surface of the water body passively. It does not have any locomotory structures. So it will act passively be carried by the water currents, and if it happens to meet the female flower, then pollination will be brought about. Okay, so that's how it happens in Vallisneria. In sea grasses like Zostera, it will happen within the water body itself. Neither the male flower goes to the is released onto the surface, or the female flower reaches the surface. Both of them are submerged within the water body itself. Pollination will happen. Yes, Yatan, you may study. Now, here you have separate male flower and separate female flower. Male flower will release the male pollen grains inside the water. The pollen grains here are long and ribbon shaped. Because of their shape, size and density, they will not rise to the surface of the water within the water body itself. They will passively float. The female flower also does not reach the surface. So, it will reach the female stigma right within the water body and bring about pollination. That's how it happens in Zostera. Okay? Next. Animal pollination or zoophily. You can find so many different types of animals involved in bringing about pollination. Some rodents, some very small primates, lizards, bees, butterflies, other flies, wasps, moths, all of these are involved in bringing about pollination. So if an animal brings about pollination, then you call it zoophily. Okay, so that is a biotic agent. In most angiosperms, animals itself will bring about pollination. Among animals, insects are the most important pollinating agents. Among insects, bees are the most dominant pollinating agents. Okay. Now, what are some of the characteristics that you can find in insect pollinated flowers? Obviously, insects are living organisms. They can see, they can smell, they can taste, they eat. So, in order to attract insects, the flowers will have certain features that are attractive. First impression, it should attract it, right? So, for that, some of the attractants that flowers have to draw the attention of the insect is color. Insects can see, so they are brightly colored. They may be large and showy, but in case they are very small, they will all, come together and form a inflorescence that is very conspicuous. Okay, so first thing, it will look good to the for the insect. Secondly, it will release a odor. It could be good odor, very fragrant, or it can be a bad odor, as bad as rotting meat or cow dung. So if it is like a very nice odor, then insects like bees are attracted to it. If it releases a bad odor, then it will attract dung beetles and flies and all of those things. Okay. I'm glad you found it useful, whoever this is. Yarini Gayatri, Nandri. Just a moment. Yaro? Just a minute. Okay. So they are clustered into an inflorescence. I've told you all that. Where was I? I'm sorry, I forgot. Yeah, I think I was saying the flowers are going to produce attractants like fragrance and color. They are colorful. That's how it will initially attract the insects or any living organism towards it. They are called attractants. Now, okay, the insect or whichever animal visited the plant, visited the flower. In order to make sure that the insect visits the flower again, it has to provide certain rewards, right? So it will reward the insect for coming to it by giving it like a little bit of snacks, a little bit of juice. Okay, so it'll give snacks in the form of pollen grains. It'll give juice in the form of nectar. So they are called rewards. Apart from that, many flowers will also allow insects that come to pollinate it to lay eggs within itself. Okay, so it'll provide a safe space for the insects to lay its eggs. All of these are rewards. Okay. And yeah, sometimes it so happens that the insects go to the flower, eat the pollen grains, drink the nectar and not bring about pollination. Such insects are called pollen or nectar robbers. Okay. Now, let's talk about examples of flowers allowing insects to lay eggs. In Amorphophallus flower, which is the tallest angiosperm flower, insects are allowed to lay eggs inside. Also, you have a plant called yucca plant which produces this yucca flower. This will allow a moth called yucca moth to lay eggs inside it. That is also like a reward that the insect is getting for bringing about pollination. Now we've learnt about the different agents of pollination. We know about self-pollination and cross-pollination. Let's now understand what something called outbreeding devices is. You know, self-pollination, if it happens continuously over a long period of time, it's not good for that particular plant species itself. Similar to how in humans, whenever very closely related individuals get married, their children will end up having, there will be a greater chances of children having genetic abnormalities, like they could be deaf and mute, or they could have some chromosomal disorders. The chances are high, that could happen to plants too. If continuous self-pollination is happening, it leads to a phenomenon called inbreeding depression, where the offspring that is formed will not be good enough. It will not grow very well. It will not reach adulthood to produce flowers. It may not reproduce very well. It will be very, easily prone to getting diseases and infections and all of that. So, uh, yeah, so that is called inbreeding depression. It happens as a result of continuous inbreeding or self-pollination. So plants know that. Plants may not have brains, but they are clever too. Every organism on this planet have one biological purpose: to make sure that the next generation is formed, they reproduce and continue to survive on this planet. Okay. So plants also, over the course of evolution, have realized that if they continue to undergo self-pollination, then it's not good for their own babies, because the babies will not be formed healthy enough. So they have come up with many devices that will kind of reduce or prevent, discourage self-pollination and encourage cross-pollination. Such devices or mechanisms are what we refer to as outbreeding devices. Vatika, I've been calling you Swastika. Okay, I'm so sorry. Sorry, Swatika. I'm a little blind in these lights. I can't see very well from that distance. Sorry. Okay. Hi, Jay. Good evening. Okay. So, inbreeding depression reduces the survival as well as fertility of offspring. So, continued self-pollination will result in inbreeding depression. So, to overcome that, plants would have evolved outbreeding outbreeding devices. I was about to say outbreeding depression. Yeah. So, outbreeding devices. There are 4 such outbreeding devices which is part of your syllabus. The first one is when there is no synchrony. Even if it's a bisexual flower, if you want to prevent self-pollination, one of the ways in which plants do that is by not producing and releasing the pollen grains and making the stigma mature at the same time. If it happens at the same time, there are mighty chances that self-pollination can take place. So in such plants, what happens is either the pollen grains are formed and released first, or the stigma matures first. Both of them will not happen simultaneously. That is one way self-pollination can be avoided. Secondly, the position of the stigma and the anther in the flower is such that even if the pollen grains are released, it's not likely for the stigma to receive the pollen from the same flower. The positions are like that. Okay. In most of these outbreeding devices, you can't 100% ensure that self-pollination does not happen at all. These plants are trying as much as they can to make sure self-pollination does not happen. Okay. Now, third one is a genetic mechanism. It's called self-incompatibility. So, if it's like the stigma receives any pollen grain having the same genotype as itself, it will realize that it's from the same plant or from a similar plant carrying similar genotype. So, it will reject it right away. Once the pollen grains falls on the stigma will not even allow the pollen grain to form the pollen tube. Okay. So if the pollen tube formation does not happen, obviously fertilization will not happen. So it will reject it right over there. It's a genetic mechanism. When we discuss pollen-pistil interaction, you will understand. So unless the stigma says yes, go ahead, the pollen grain will not be able to form the pollen tube. See, even plants understand that no means no, but humans will not understand. So if the stigma does not allow pollen grains to form a pollen tube, it will be rejected. It will only allow pollen grains that have a different genotype than itself to form pollen tube. So that genetic mechanism is called self-incompatibility. If the pollen grain from the same plant, self plant, falls on the stigma, then it is not compatible. It is incompatible. So it will be rejected. Okay. All right. Now, last one is production of unisexual flowers. This means that flowers either have only androecium or gynoecium. They are either male flowers or female flowers, but they are not bisexual. In plants like maize and castor, you have unisexual flowers produced within the same plant. The plant is monoecious. There's only one plant. The single plant will produce separate male flowers, separate female flowers. In that case, you can avoid autogamy, but you cannot avoid geitonogamy. In case of flowers like papaya flowers, where there's a separate male plant, separate female plant, the plant is dioecious. In such cases, autogamy, geitonogamy, both can be avoided. Okay, so that is outbreeding devices. Now let's talk about pollen-pistil interaction. See, in nature, many flowers produce pollen grains at the same time. In nature, you don't have only one species. It's not like all the rice plants in the world is inside one chamber and the rice plant's pollen grain is going to land on the rice plant stigma itself, right? So, it's possible that many different pollen grains can land on many different stigmas. If two plants, two different species plants are growing right next to each other and both of them end up producing pollen grains at the same time, becoming stigma, becoming receptive at the same time, the pollen grain from a hibiscus plant could land on the stigma of the jasmine plant that is, that is lying right next to it. So it's possible. But any plant's pollen grain cannot form a pollen tube on any plant stigma. Who decides which pollen grain forms the pollen tube? That is decided by the stigma. Once pollination happens, the stigma and the pollen grain will communicate, not by words but by means of chemicals. There are certain chemicals present on the surface of the stigma that will communicate with the chemicals present on the surface of the pollen grain. So a chemical dialogue happens, and unless the stigma is sure that this pollen grain belongs to my same species, this pollen grain is compatible, it will not allow it to form a pollen tube. Okay, so that is called pollen-pistil interaction. So based on all of these factors, the stigma will either accept the pollen or reject the pollen. If it accepts the pollen, it will allow the pollen grain to form the pollen tube. If it rejects the pollen, it will not allow it to form the pollen tube. Okay. Now, yeah, this is called pollen-pistil interaction. So what is pollen-pistil interaction? From the time the pollen grain is released and gets deposited on the stigma to the time it enters the ovule, all of the events that take place is called pollen-pistil interaction. Here you can see many pollen grains have landed on the stigma, but only some of them are allowed to form the pollen tube, not all of them. Because the ones that are not allowed to form the pollen tube are probably from a different species or same species but incompatible. It's not allowed to form a pollen tube. Okay? Yeah. So that is pollen-pistil interaction. It falls here. If it is compatible, it will be allowed to form a pollen tube. The pollen tube penetrates the style, goes into the ovule via the micropyle and then release its contents inside. So after successful pollination, what is the next step? The pollen tube enters the embryo sac within the ovule. See, first it lands on the stigma. Through the germ pore, the pollen tube will form. The pollen tube is formed from the vegetative cell. Okay, so that will elongate and form a pollen tube. It will, take in nourishment from the stigma where there are sugary substances. Through the style, it will absorb nourishment and it will grow deeper and deeper inside the style. And eventually, it will enter the ovule through the micropyle. Once it has entered the ovule, it will enter the embryo sac also. It will enter the synergids of the embryo sac. Within the synergids of the embryo sac, there are cellular thickenings that are finger-like, which are called filiform apparatus. This will kind of guide the pollen tube. So once the pollen tube enters there, it will release its contents inside. Within the pollen tube, you will have two male gametes, right? So within this, you will have one and two male gametes. Both of these male gametes are released inside. This is one male gamete, second male gamete. The first male gamete will fuse with the egg cell to form the zygote. The second male gamete will fuse with the polar nuclei to form what is called the primary endosperm nucleus. Okay, see here, it will come through this, enter inside, and it will fuse. Since here, one male gamete is fusing with this, the second male gamete is fusing with this, two fusion events are happening. We call this double fertilization. Okay. Okay, Swatika. Which class? You mean these sessions? You want me to call out names of regular students? All right, so we'll move on. So this is double fertilization. The first fusion event is the haploid male gamete fusing with the haploid female gamete to become the diploid zygote. This zygote will eventually give rise to the embryo. Embryo becomes the future plant. Where is all of this happening? It is happening inside the ovule. Remember that. The next fusion event is called triple fusion because three nuclei are fusing here. One male gamete and two polar nuclei to form a triploid structure called primary endosperm nucleus. This will become the endosperm in future. The endosperm nourishes the developing embryo. Zygote has to develop from the embryo, develop to form the embryo. Zygote will undergo mitotic divisions to give rise to the embryo. Primary endosperm will undergo mitotic divisions to give rise to the endosperm. Initially, both of these are unicellular structures. They will undergo mitosis to become multicellular. Now, for zygote to develop into the embryo and the embryo to develop into its successive stages, nourishment is required. That nourishment is provided by endosperm. If endosperm doesn't exist, this development cannot happen. So, endosperm development happens first. After some amount of endosperm is produced, that can be used as a source of nourishment for the development of the embryo. Though zygote formation, primary endosperm nucleus formation happens simultaneously, the development of endosperm precedes or happens sooner compared to the development of the embryo. That is because for the development of embryo, at least some amount of endosperm is essential. Okay. Now, let's talk about the endosperm. Like I already mentioned, it's triploid. It is nutritive. It is used in the nutrition of the developing embryo and it is filled with reserve food material. So when endosperm is developing, what happens is similar to how when female gametophyte was formed, initially free nuclear divisions happened, later cell walls were laid. Even over here, initially the cell division will be free nuclear. So the nuclei will keep on dividing and form a free nuclear endosperm, no cell walls between the nuclei. Later, cell walls will form and it will become the cellular endosperm. To give you an example, in tender coconut, the tender coconut water that we drink, that is the endosperm. The water is free nuclear endosperm where free nuclei are present, there are no cell walls between them. And in cellular endosperm, cell walls would have already formed. So the kernel of the coconut, the white color part of the coconut that we grate and make, cookies and sweets and other dishes out of, that is called the cellular endosperm. Okay. Is that clear? I hope it is. Now, all of this is present within the seed. Endosperm is also located within the seed. Once the seed, matures, sometimes endosperm will be fully consumed. Endosperm will no longer be left because during embryo development, endosperm would have gotten used up. And in some plants, you can find endosperm still being present. Depending upon whether the endosperm is still present or not, you have two different types of seeds, endospermic and non-endospermic. Okay, so in case of peas, groundnut and bean, endosperm is fully consumed. And in case of castor, maize, coconut, endosperm is retained even in the mature seed. Okay. Thank you very much, Sneha. May God bless you. Get well soon. Now let's talk about the embryo. Embryo develops from the zygote, like I told you. Zygote is a single diploid cell that is formed. This zygote will eventually give rise to the future plant. Okay, remember all of this is happening inside the ovule, inside the seed. So once you sow a seed inside the soil, a tiny plant will germinate from there, right? That tiny plant that germinates is formed from the zygote. Zygote first forms the embryo, and from the embryo, that plant will germinate. The germinated plant will form. Now, here the zygote will undergo certain developments during embryo formation. The single diploid cell is called zygote. This zygote will undergo multiple mitotic divisions. And first, there is a phase called the globular embryo stage, where the embryo is clearly globular. In this stage, you can also find what is called the suspensor. This globular part here is what is going to form the future plant. So this is called the embryo proper. And here you have the suspensor. The reason the suspensor is there is to draw nourishment from endosperm. See, this is the seed. Within the seed, you have the endosperm. Within the seed, you have the embryo. For the embryo to develop further, nourishment has to be drawn from the endosperm. So the suspensor part of the endosperm suspensor part of the embryo penetrates into the endosperm, and from the endosperm it will draw nourishment and use that nourishment to develop further. This globular embryo, after a period of time, will start appearing heart-shaped, the embryo proper. So that is called the heart-shaped embryo, and eventually this will form the mature embryo. The early embryonic stages in both dicot and monocot plants are similar. Okay? Hi Shagun, good evening. Now, what you're looking at here is a mature dicot embryo. There are some differences in the dicot embryo and in the monocot embryo. We'll learn about that. We'll start with the dicot embryo. As the name itself indicates, dicot means having two cotyledons. Okay. So in all embryos, you will have one embryonal axis. Okay. So this is the embryonal axis. To this embryonal axis, the two cotyledons will be attached. There is a point on the embryonal axis where the two cotyledons will be attached. This point is called the cotyledonary node. The portion of the axis above the cotyledonary node is called the epicotyle. Epi, above, cotyle, the position of cotyledons, epicotyle. And the portion of the axis below the cotyledonary node is called the hypocotyle. The tip of the epicotyle is called the plumule, and the tip of the hypocotyle is called the radicle. These are the parts. I have shown you this elongated embryonal axis, but actually inside a seed, within the embryo, you do not find such a long axis. If you've seen these dicot plants, like your sprouts, if you've seen the sprout of green gram, you will notice how there are two cotyledons, but there is this small line that arises, right? So that is the embryonal axis. Okay, so it's not always so long. For your understanding of different parts, I've made it very long, but actually it will be very small. So in this dicotyledonous embryo, this is the cotyledonary axis, embryonal axis. This is the cotyledonary node. Here is your first cotyledon. Here is your second cotyledon. The region of the axis above the cotyledon is called the epicotyle. The tip of that is called the plumule. The region below the point of attachment is called hypocotyle. The tip of that is called the radicle. So this will have a root cap. The radicle in future is going to give rise to the root system. The plumule is going to give rise to the shoot system. Okay. All right. Next, this is the dicot embryo, and here we have the monocot embryo. Many structures are similar, but here additionally you will have two things. We'll talk about that. Here you have the here you have the embryonal axis, and this is the cotyledonary node. To the cotyledonary node, you have one cotyledon attached because this is a monocot embryo. There's only one cotyledon. The single large shield-shaped cotyledon is called the scutellum. This is the epicotyle, this is the hypocotyle. To the epicotyle, at the epicotyle tip, you have the plumule or the shoot apex. At the tip of this, you have the radicle. Below the radicle, you have the root cap that is protective in nature. This is common to both that and this. There are three new structures that you will notice here. One is coleoptile, epiblast, coleorhiza. What is epiblast? Let's understand. In dicot plant, at the cotyledonary node, you had two cotyledons, but here you have only one cotyledon. The remnant of another cotyledon is called the epiblast. Okay. So this other cotyledon is highly reduced here. What is coleoptile? A sheath-like covering for the plumule is called the coleoptile. It's protective in function. The sheath-like covering for the root or the radicle is called the coleorhiza. That is also protective in function. Is that clear? Is that clear? Is there any doubt between the monocot and dicot embryo? What time is it today? 5:42. Any doubts, boys and girls? Okay. We'll move on. Now let's talk about artificial hybridization in case you want to produce a hybrid between two varieties of the same species, let's say. One plant has a very high yield but very low tolerance to pest infestation. Another plant has very low yield but is highly pest resistant. You want to bring the high yield character of one plant and the high pest resistance character of the other plant within the same hybrid. Okay, so you want to cross pollinate these two plants. So what do you do then? If it is a bisexual flower, then among both these plants, one of the plant has to be designated as female. Let's say the plant that produces high yield, that gives a high yield, let's assume that plant as female. Let's designate that plant as female. If I'm calling it a female, then I should remove the male parts from it, right? So what will I do in this bisexual flower? I will cut out the male part before it matures. Even before anther matures, even before it produces pollen grains, I will cut it. Why should I do that? Because I want to cross-pollinate it with another plant. I do not want self-pollination right now. To prevent self-pollination, you will remove the male part of the flower before it matures. That process is called emasculation. You do that if it is a bisexual flower. If the flower itself is unisexual, then it will either have only pistil or only have stamen. So if it's unisexual, that step is not necessary. But if it's a bisexual flower, then whichever plant you've designated as the female, from that plant you should cut and remove the anthers before maturity. Okay. First step, emasculation. The second step is you have made sure that the same plant's pollen will not fall on it. Same flower's pollen will not fall on it by cutting it. But you should also ensure that some random pollen grain should not come and get deposited on that. So you will tie a bag around it. This process is called bagging. Bagging will prevent random pollen from contaminating the surface of the stigma. What do you do next? You will take pollen grains from your choice of plant, whichever has your choice of characters. You will take that and go dust it on the stigma of the female flower. This way you have ensured that the right kind of pollen has been dusted on the stigma. After this, you will tie a bag around it again to make sure that some random pollen will not come and get deposited there. After that, fertilization will happen, fruit formation, seed formation, all of that will happen. Within the bag itself, the fruits will form, seed will form. From that, you can collect the seeds, sow them in the soil again and check for which plant has your desired combination of characters of high yield and high pest resistance. Okay. So these are some of the steps. Firstly, you have emasculation, where masculine is male. Emasculation is removal of the anther before it dehisces. Required only in bisexual plants, not needed in unisexual plants. Plants producing unisexual flowers. Then bagging to prevent contamination, dusting of pollen. Again rebagging after dusting, and then you will allow the fruits to develop within the bag and collect the seeds later. Okay, just a minute, I'll be back. Okay, we're back. Where can you find the notes? What is your name? Mecklin God Pretty. You can find the notes on our WhatsApp channel. The link to our WhatsApp channel will be pinned in the chat or it will be given in the description box. One of those. So you can check it out there. Okay. All right. So we're done with artificial hybridization. We'll move on to seed. Seed is the final product of sexual reproduction. Seeds are like babies for plants. What are the different parts that you can find in a seed? How do seeds form first of all? The ovules present inside the ovary, once fertilization happens, ovule itself will transform into the seed. The ovary wall will transform into the fruit. Okay. So it's not wrong if you call seed as a fertilized ovule. So this is fertilized ovule. This is formed inside the fruits. What are some of the typical parts of a seed you can find? Seed coats. Within a seed, you will have seed coats. How did the seeds form? It formed from the ovule. Ovule had ovule had integuments, right? Outer integument and inner integument. The integuments of the ovule gave rise to the seed coats. That itself transformed into the seed coats. The integuments were protective of the embryo sac. The seed coats are protective of the embryo. Okay. Apart from that, depending upon whether it's a monocot plant or a dicot plant, you will have either one or two cotyledons and you will have the embryonal axis to which the cotyledons are attached. Now, depending upon whether at maturity the seeds have endosperm or not, you can classify seeds into two types: endospermic seeds or albuminous seeds and non-endospermic seeds or exalbuminous seeds. Now remember this, every seed will have endosperm present in it when uh, double fertilization happens. Endosperm will 100% form in it. But the question here is whether endosperm will persist in the seed till the seed becomes mature. Will the embryo consume the entire endosperm during its development, or some amount of endosperm will still remain? That's the question, right? So, based on the presence of endosperm in a seed at maturity, you have these two different types of seeds. One is non-albuminous seed, the other one is albuminous seed. In non-albuminous seed, residual endosperm will not be present. Examples for that includes pea and groundnut. In albuminous seed, some amount of endosperm will still be present inside the seed. Example for that is wheat, maize, barley, castor. Okay. Now, there's another thing called perisperm, which you can find only in some seeds. Sperma means seed, peri means around. Perisperm is the nucellus, the residual nucellus that you can find in seeds even at maturity. Normally what happens after double fertilization is most of the cells, other cells of the embryo sac like the antipodal, synergids, all of them will disintegrate, degenerate. The nucellar cells also will start getting used up. So in the mature seed, you don't get to see the nucellar tissue. But in case of some species like beet and black pepper, even in mature seeds, you can find some amount of nucellus still being present. So that is called perisperm. So it is the residual persistent nucellus that you find even in mature seeds. Examples for that includes black pepper and beet. Next, what are some advantages of seeds in angiosperms? So firstly, in angiosperms, you have fruits along with seeds. In gymnosperms also seeds are produced. But the seeds in angiosperms are very different from the seeds in gymnosperms because in angiosperms around the seed you have fruits, right? So because of the presence of fruit wall around the seed, there are many mechanisms for dispersal of seeds. These fleshy fruits will act as a source of nutrition for animals that come and feed on it. Once the animal is feeding on the fruit, it will end up feeding on the seeds as well, and the seeds will be dispersed wherever the animal decides to excrete. So from where it is formed, it can go be dispersed in different areas and occupy new habitats. And within the fruit and the seed, there are sufficient food reserves until the seed germinates and gives rise to, produces a shoot system, starts photosynthesizing, making its own food. It should have sufficient nourishment that is available in the form of either cotyledons or in the form of endosperm in case of angiosperms. So that is another advantage. Thirdly, they have a hard protective seed coat which will protect the uh, embryo that is lying inside. Next, because this is a product of sexual reproduction, newer genetic variations will arise, newer genetic recombinations will happen. Seed is the basis of agriculture. Many crop plants are produced by using seeds only. So you may have seen how ragi, maize and uh, 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 rice, all of these crops are grown. Many crops are grown by making use of seeds itself. And in seeds, as they mature, they lose a lot of water. Okay, so they become more dehydrated and they become dormant. Unless they are in moisture and the right kind of environmental conditions are available, they will not germinate. Because of this property of dormancy, it's possible for us to store seeds for a long period of time. We can store rice grains in our homes to cook rice. We can store wheat. We can store olive oil pulses. All of them are seeds, right? What if the seed immediately after it formed started to germinate? We couldn't have stored them. Because of the seed's property of dormancy and dehydration, we are able to save them and store them as food and use them as food grains throughout the year. So that is one advantage, right? And yeah, that's how even farmers can store these grains and seeds and use them for the next cycle in their agricultural fields. So that's the story about seeds. Similar to how pollen grains have a viability period, seeds also have a viability period. For as long as the seed is capable of germination and producing a new plant, we say that the seed is viable. This viable period varies again from species to species. In some of them, it could be just a few months. In some of them, it could be many years. We have some examples of plants in which the seeds, even after thousands of years, have retained their viability, not just germinated but also produced flowers. So here you have the example of Lupinus articus. This was found, the seed of this Lupinus articus, which was 10,000 years old. It was dormant for 10,000 years. It was found during an excavation uh, in the Arctic tundra. It was found within the ice. It was stored there. So that's where it was found. And after being found, when the right conditions were provided to it, it germinated. Not only did it germinate, it also produced many flowers. Okay. So its viability period was as long as 10,000 years. There's another example called Phoenix dactylifera, which is nothing but the common dates that we eat. This was also found during the excavation. It was 2000 years old, and it was found during the excavation of, at a site near King Herod's palace, which is located close to the Dead Sea. Even though it was 2000 years old, it was still viable. It did germinate and give rise to a new plant. Okay. Now, yeah, it's the date palm. Iftar ka time ho gaya. Iftar means your food, like you consume food after sunset or something, that one. Okay. All right. So that's about seed viability. These are two examples that you must remember. Next part, we have fruit. We are almost reaching the end of the chapter. What is fruit? Fruit is the ovule. After fertilization, the ovary transforms into the fruit. So the fruit has two parts. It has seeds inside and it has the fruit wall. The wall of the fruit develops from the wall of the ovary and it is called the pericarp. Peri means around, carp means fruit. So here the fruit, depending upon the nature of the pericarp, whether the pericarp is fleshy or dry, you have two different types of fruits. One is the fleshy fruit, one is the dry fruit. The dry fruit here is not like the dry fruits that we commonly use, like we call cashews and badam as dry fruits, right? And raisins as dry fruits. That's a different kind of dry fruit. Botanically, uh, excuse me, a fleshy fruit and dry fruit are different from each other because the wall of the fruit is either fleshy or dry. Okay. So fleshy fruit examples include guava, orange and mango, and dry fruits it is groundnut and mustard. You must remember examples for everything. Just give me a minute. Yeah. So that's about these classification of fruits. Another way of classifying fruits is based on which part of the flower produces the edible part of the fruit. We consume many fruits, right? We consume certain parts of the fruit. If the part of the fruit that you are consuming, the edible part of the fruit, if it arises from the ovary, then you call it a true fruit. If it arises from any other part other than the ovary, then you call it a false fruit. In case of apples, in case of uh, uh, strawberries, in case of cashew, the part of the fruit that we consume is not the ovary, it is the thalamus. So such fruits are called false fruits or pseudocarps. Okay. Now, here you can see the central hard part of the apple that we throw away. That is the actual ovary of it. Surrounding that, whatever part we consume, that is the thalamus part of it. Even in strawberries, the fleshy part that we consume is the false fruit. It is the thalamus. The small grainy structures like dots that you can find on strawberry, they are its actual fruits and they are called achenes. Okay. Now, let's talk about parthenocarpic fruits. Normally fruits are produced after fertilization. It's a product of fertilization. Following fertilization, ovule will develop into the seed, ovary will develop into the fruit. But sometimes, even without fertilization, fruit can form. If fruits are forming without fertilization, we call them parthenocarpic fruits, and such fruits don't have seeds. They are seedless. In banana, these fruits are parthenocarpic naturally, okay? But sometimes by applying certain hormones, plant growth regulators, you can induce parthenocarpy in certain plant species, okay? So it can be induced or it can be natural too. If you remember, when you learnt about auxins, auxins are plant growth regulators. So in auxins, when you spray them on tomato plants, the tomato plants become seedless, right? The fruits will become seedless. So it induces parthenocarpy in tomatoes, we've learned. So that is one example. Next, we have something called apomixis. Normally, seeds are formed after fertilization. It's a product of sexual reproduction, but in apomixis, even without fertilization, seeds will form. Okay. So when you look at a seed, you mostly think that it is a product of sexual reproduction. So a characteristic feature of sexual reproduction is seed formation. If asexual reproduction happens by formation of seed, then you call it apomixis. Okay. See, this is some sort of mimicry. Like normally, what do mimicry artists do? You may have watched this Kapil Sharma show where Sunil Grover impersonates a lot of different people. So every time he's trying to act like Amir Khan, he acts or he imitates certain characteristic actions of Amir Khan. If he does that with Shah Rukh Khan, he imitates certain characteristic attributes of Shah Rukh Khan. Like that, in apomixis, there are certain plants that are undergoing vegetative reproduction or asexual reproduction, but they are acting like sexual reproduction. How does it act like sexual reproduction? By mimicking some aspect of sexual reproduction. Which aspect of sexual reproduction? Seed formation in sexual reproduction is what they will copy. Normally, seed formation will happen during sexual reproduction, but these guys will produce seeds even without sexual reproduction. So, that is asexual reproduction pretending to be sexual reproduction. That is called apomixis. This happens in plants such as Asteraceae members and grasses. So it's a special mechanism that produces seeds without fertilization. If fruits are formed without fertilization, you call it parthenocarpy. If seeds are formed without fertilization, you call it apomixis. Okay. So apomixis is a form of asexual reproduction actually, but it's acting like sexual reproduction by producing seeds. Okay. But in those seeds, formation and fusion of gametes would not have occurred. So how do apomictic seeds develop then? So here what happens? The egg cell is diploid. Normally, how does the egg cell form? From the megaspore, it will undergo mitotic cell division and form. But for megaspore formation, meiotic division should have taken place. But here what happens is, in the nucellus, one diploid cell will directly act like the egg cell, and that itself will yeah. So that itself will develop into the embryo without fertilization, and that's how you will get an embryo inside a seed. These can be beneficial to farmers if they are, looking for hybrid seeds, looking to buy hybrid seeds every year. If they can make the hybrid plants apomictic, then year after year they need not bind. They need not buy hybrid seeds from the market. That will save them a lot of money. Okay. Now, let's talk about polyembryony. It's in the name itself. Poly means many. Embryony refers to embryo. Normally, within one seed, you will find only one embryo, but within one seed, if you have many embryos present, more than one embryo present, then you call it polyembryony. So, this can be seen in plants such as citrus and mango, where within one seed, you can find many embryos. So, when you sow that one seed, many plants will start germinating from it. Okay. So, here what happens is you will have one normal embryo that's produced from the zygote. Apart from that, certain nucellar cells or integument cells will start dividing and acting like embryos and penetrate into the embryo sac, and they will develop into embryos. So that's what will happen here. That is polyembryony. With that, we have finished sexual reproduction in flowering plants. Is it clear so far? Verma is asking for question practice. In today's session, question practice is not going to happen. Question practice will happen in the upcoming sessions. Once this entire theory series, revision series will get over, following that you will have question practice. Is it clear? All right, we can get started with principles of inheritance and variation. If everybody is ready, give me a thumbs up in the chat. Okay, so we'll move on. So what is principles of inheritance and variation chapter? We will learn about the basics of genetics in this chapter. As the name itself indicates, we will learn about what are some of the principles that will govern inheritance, what are some of the principles that will govern variation. So what is genetics first of all? This is the first chapter in the genetics unit. So genetics is one branch of biology, just like you have physiology, anatomy, psychology, many branches of biology. You have genetics also as a branch of biology. In this branch, you will learn about inheritance and variation. Now, what is inheritance? Inheritance is the process by which characters are passed on from one generation to the next. Variation is the basically the degree to which parents differ from their offspring and also offspring differ from each other within the same generation. The difference between siblings is what is called variation. Even though they are born to the same set of parents, they don't look identical unless they are monozygotic twins, right? So differences will exist from parent to offspring and between offspring also. So that difference is what we call variation. All right. So now let's understand the history of genetics. Since very early on, when humans stopped their hunting and started to settle down, human civilizations began. People started to rear animals that were important for them. People started to grow plants that could act, I mean, that could be used as food. They started rearing crops and animals and all of that. So that's when humans kind of had an idea that in all living organisms, variation exists. It could be plants, it could be animals. Not all cows, though they belong to the same species, will produce the same amount of milk. There is variation even over there. So they knew that variation existed, and they even knew that the cause for this variation is sexual reproduction. So they tried to use that to their benefit. So if they wanted cows that produced more quantity of milk, they took cows that they had which produced high yield of milk and crossed them with bulls that were more hardy and could work hard in the fields. And they tried to artificially select um and produce such hybrids even back then. One such hybrid variety of cow that we have been able to obtain is called Sahiwal breed of cow in Punjab. So our ancestors knew about this. The proper scientific basis for genetics was established because of uh, Mendel's experiments. Mendel performed experiments using pea plants for 7 years, starting from 1856 to 1863. He was a he lived in a monastery. He was a priest, and in his monastery garden, using the pea plant, he performed many experiments for a duration of 7 years. And based on his observations of experiments, he proposed what is called the laws of inheritance. Back then, they were called just principles, but later we called them the laws. Okay, we gave them the position of laws. They are called the laws of inheritance because he's contributed so much to fundamental genetics. He's called the father of genetics. While he performed experiments, he had a large sampling size, meaning at a time he used to work with like thousands of plants. Because he had such a huge sampling size, there was more credibility to his data, to his results. And whatever data or whatever results he obtained after his crosses, after his experiments, Just after one set of experiments, he did not announce his observations, he did not publish his results. He tested that for the successive generations also, and only after time he got similar results did he publish his findings in science journals. And he is one of the very few biologists to have integrated mathematics and biology. He applied statistical analysis and mathematical logic to solve whatever problems we had in biology. So that's about Mendel and his contribution. The reason why he chose the pea plant is because we are learning about genetics and you do not want one generation and next generation to appear after 30 years, 20 years or 5 years. You want the next generation as soon as possible. Since pea plants have a short lifespan, they will germinate, the seeds will germinate and vegetative growth will happen. It will produce flowers also and produce fruits also within a span of maybe 3 to 6 months. So since you're getting the next generation very quicker, this is ideal for genetics experiments. Secondly, this is not very difficult to grow. These are small climber plants. Very little labor is required. In a very short, very small space, many plants can be grown. Next, within pea plants, there is variety of visible characters, which means they are different. They show variety in their height with respect to how tall they can grow, what color flower they produce, what color seed they produce, what is the shape of the seed and all of that. So there are visibly differing characters that are present in pea plants, which is what is very essential for genetics experiments, since we're trying to understand how one character is passed on from one generation to the next. Next, each fruit will produce large number of seeds. One plant will produce many fruits. So from one parent plant, you get many, many little babies. So you have more credibility to your data. And pea plants are easy because the pea flowers, if you leave it undisturbed, you don't touch it, meddle with it, it will undergo self-pollination. But if at all you want to cross-pollinate it, these bisexual flowers, they're easy to cross-pollinate. So these are some of the advantages. So Mendel, what he did in his experiments was he chose 7 different characters in pea plant. And each character had 2 opposing traits. For example, height of the stem is one character that he chose. And height of the stem can either be very tall or it can be very dwarf. It can either grow as tall as 6 feet or it can be under 2 feet in height. So there's no in between. 2 contrasting traits, we call that. So he chose 7 such characters. Each character had 2 contrasting traits. So totally he chose 14 such varieties of plants. These are the characters that Mendel chose. The first one is the height of the stem. There's one character for stem, which is height. It can either be tall or it can be dwarf. This is the character. These are the traits. Then there is 2 characters for flower, the color of the flower and the position of the flower. The flower can be violet or white. The position can be axial or terminal. And the pod color and pod shape. Pod color can be, pod is the fruit of the pea plant. Pod can be green or it can be yellow. Pod can be inflated or it can be constricted. Speaking of the seed, seed has 2 characters, seed color and seed shape. Seed can be yellow or green. It can be round or wrinkled. Okay. So these are the 1, 2, 3, 4, 5, 6, 7 characters and 14 traits that Mendel chose. Next. Here is a list of the same, a table of the same. Now, Mendel conducted such experiments using several such plants, 14 such varieties of plants. I want you to please stop talking among yourselves and pay attention. If you're not interested, you may leave. Please stop spamming and conversing among each other in the chat. All right. So he chose several such true breeding varieties of plants. 14 such varieties of plants was chosen by Mendel. Uh, in his experiments, he performed three very important steps. Firstly, he selected parents. The two parents that he selected for these plants, yeah, they were different with respect to one trait. If one of them was tall, the other one would be dwarf. If one of them produced purple flowers or violet flowers, the other one would produce white colored flowers. So the parents showed contrasting traits. So firstly, he selected such parents and he made sure that those parents were homozygous. They were pure breeding or true breeding types, which means if you self-pollinate them and if they produce seeds, they will all be true to their parental type. So that's what it means. So he selected such plants and then he performed cross-pollination between them. That is the next step. Just give me 1 minute. All right. Cool. So he performs experiments where he selects plants like that. And first he allows the two plants of contrasting traits to undergo cross-pollination. Obviously, how cross-pollination is done, you know. You designate male and female plants, you do emasculation, you do bagging, transfer the pollen and all of that. And whatever plants, whatever seeds he obtains from that cross-pollination experiment, when the seeds are sown, whatever plants you get in the next generation, that is called um, first filial generation or F1 generation. Okay. So this F1 generation of plants is allowed to undergo self-pollination next. After they are self-pollinated, whatever offspring they get is called the F2 generation. So he will do all of these steps in his experiments and analyze what kind of offspring he obtains in the F1 generation, what kind of offsprings he obtains in the F2 generation and all of that. So for example, let's say in his experiment called Monohybrid cross, at a time he will choose only one character. He will take the two parents that are of true breeding type. They have contrasting traits. For this time, let's assume the character that he chose is the height of the stem. One plant is tall, one plant is dwarf. He performs a cross between these two. When he obtains the F1 generation seeds, he sows the seeds and he ends up getting all tall plants in the F1 generation. After F1 generation plants are self-pollinated, he obtains what is called the F2 generation. Okay. So in the F2 generation, he will notice that 75% of the plants are tall and 25% of the plant is dwarf. Okay. So based on his observations here, he will infer certain things. First of all, what are the observations that he made? In the F1 generation, when you cross tall plant with dwarf plant, all of the plants appeared like one of the parent only. None of the plants were dwarf. Okay. So this is the F1, this is one observation that he made. Secondly, in the F2 generation, both of the traits appeared in 3:1 ratio. Here the plant was not dwarf. Male and female both were tall. We are self-pollinating here. But if you look at the F2 generation, there is a dwarf plant which resembles like the parental generation, which is not there in its immediate parents. So this is something that he will notice. And he will also notice that either in the F2 generation or in the F1 generation, the plants did not show any blending. Blending means either the plant is tall or it is dwarf. It's not intermediate in height. Okay, so he notices that. What he infers from this is there is something that is stably being passed from one generation to the next generation, and this is being passed down via the gametes. Okay, and he calls these stable things as factors. Today we call them as genes. We know that genes are the units of inheritance. If any character has to pass on from one generation to the next, it happens via gametes through the genes. And different genes, we have many different genes present in our body. Each gene can exist in more than one type. Genes are nothing but DNA. So the DNA sequences can vary from one individual to another. So different forms of the same gene are what we refer to as alleles. This is one thing. Next, when he performed similar experiments for all other traits, all other characters, okay, monohybrid crosses when he performed, he also noticed every time in the F1 generation only one of the two traits were visible, the other trait was not visible, and whichever trait was visible in the F1 generation, he called that the dominant trait. Whichever trait was not visible in the F1 generation, he called that the recessive trait. Okay. So when he performed experiments for all the 7 characters that he had chosen, he found out which trait is dominant and which trait is recessive. When it comes to height, tall is dominant. When it comes to flower color, violet is dominant. Flower position, axial is dominant. Fruit color, green is dominant. Fruit shape, inflated is dominant. Seed color, yellow is dominant. Seed shape, round is dominant. Okay. The others are recessive. Now, based on his observations here, he proposed what is known as the law of dominance. Just give me. Okay. So he proposed what is called the law of dominance. The law of dominance has 3 statements. Firstly, all of the characters in an organism is controlled by discrete units called factors, and these factors occur in pairs. Whenever there is a pair of dissimilar factors, heterozygous condition, whichever one expresses itself is called the dominant trait, the one that does not express itself is called the recessive trait. Now, one gene inheritance, in order to understand what exactly is happening at the cellular level, what exactly is happening with the genes, we will try and represent tallness with capital T and dwarfness with lowercase t. Since pea plants are diploid in nature, they will have 2 copies of each gene, one received from the mother, one received from the father. So the tall plant, being homozygous, will have the genotype capital T, and the dwarf plant will have the genotype lowercase t, lowercase t. Okay, so what's the first step that they will do? They will, what Mendel did was, he crossed a tall plant with a dwarf plant. So you have capital T, lowercase t, lowercase t. When it produces gametes, since it is homozygous, only one type of gamete is produced. You will end up getting all tall plants in the F1 generation that are heterozygous. When you cross them, when you self them, next, on selfing, you will obtain the F2 generation. In the F2 generation, you will notice how, because the parents are heterozygous, you will get 2 different types of gametes. When you plot that in a Punnett square, you will notice how you will get 3 different types of phenotypes, 2 different types of phenotypes and 3 different types of genotype. The phenotype is tall, 3 of them are tall, 1 is dwarf, and the genotype is capital T, homozygous dominant, 1 of them, heterozygous dominant 2, homozygous recessive 1. So the phenotypic ratio will be 3:1 and the genotypic ratio will be 1:2:1. Okay? Now, based on his observations here, he will propose what is called the law of segregation. And the law of segregation states that even though the parents contain 2 alleles, because it's a diploid organism, even though the parents contain 2 alleles, during gamete formation, the factors or alleles segregate such that a gamete receives only one of the 2 factors. This is called the law of segregation. This is a universal law. In all diploid organisms that will undergo sexual reproduction during gamete formation, even though we have 2 alleles for each gene within our cells, only one of these alleles will get passed on into the gametes. That is what law of segregation says. It's true for us also. We are diploid organisms. Every gene we have two copies, but when gamete formation happens, meiosis takes place and haploid cells are formed. So the haploid cells will receive only one of the 2 alleles that we have. That is law of segregation. Next, there is something called monohybrid test cross. So if a plant is showing the dominant phenotype, you're not sure about its genotype, it's showing the dominant phenotype, you want to identify what is its genotype, whether it is homozygous or heterozygous. How do you do that? You can cross it with the recessive plant. Okay, recessive is always homozygous, recessive cannot be heterozygous. So let's say you have a violet color flower producing pea plant. You want to know whether that is homozygous or heterozygous. What you can do is you can cross that with a white color flower producing plant. Okay. So if it is homozygous, then all of the offspring of this cross will be violet in color. If it is heterozygous, you will get 50% violet flower producing plants and 50% white color flower producing plants. Now, the law of dominance is not true all the time. There are many deviations to Mendel's law of dominance. Mendel said that every time there is a pair of dissimilar alleles, only one of that will express itself, the other one will not, right? Many people try to replicate the same experiment that Mendel did with pea plants, and some of them try to do that with a plant called snapdragon or antirrhinum. So when they performed the same steps as Mendel's experiment, what they noticed was the character that they considered here was the color of the flower. When they performed the same experiment, they noticed that sometimes the law of dominance did not apply at all. They considered a red color flower as one parent and white color flower producing plant as another parent. According to Mendel's law of dominance, in the F1 generation, all the plants should have been either red or white. Whichever one was dominant, that should have expressed itself. But here, surprisingly, they got pink colored flowers. Pink is not like either of the parents. Neither of the parents are pink. One parent is red, one parent is white, but the offspring ended up being pink. Okay, so here in the F1 generation, we got a brand new phenotype which was nothing like the parents, and this brand new phenotype was an intermediate phenotype. They continued the experiment. They allowed the pink colored flowers to undergo self-pollination, and in the F2 generation, they got the genotypic ratio and phenotypic ratio as 1:2:1. So they got one red color flower producing plant which had capital R as the genotype, and the other one, wait, this should be 50%. 25% of them were red, 25% were white, and 50% were pink. Okay, this is what they got. And if you plot, see, this is what the actual antirrhinum flower looks like. This is the red flower, this is the white flowers, these are the pink flowers. And if you, use the genotype to represent that, you will notice how every time it is in heterozygous condition, it will be pink in color. If it is homozygous recessive, it will be white. If it has both dominant alleles, then it will be red. So this is called incomplete dominance. Of the 2 alleles, neither of them will completely dominate over the other. So you end up seeing some blending. You will end up finding an intermediate phenotype appearing in the F1 generation. That is incomplete dominance. So what does dominance actually mean? You know how our DNA has genes present in it? Within our chromosomes, DNA is there. Within DNA, nucleotide sequence is there. These nucleotide sequences are nothing but our genes. So in individuals, we can have both genes of the same type, or we can have both alleles of different types. Whenever both are alleles, for each gene we have two copies because we are diploid. If both of them are the same copy, we call it homozygous. If they're both different, you call it heterozygous. How do we end up getting so many different copies for one gene? That happens because of mutations. So sometimes the normal gene, whatever nucleotide base pair sequence will be there, that will get mutated and changed. So when that happens, you get newer and newer variants of your genes. So if at all, because of that mutation, let's say there was a normal gene and that normal gene is responsible for the production of an enzyme, and that enzyme is involved in the formation of a product. if the gene is normal, if that allele is normal wild type, it will do its job. Let's say it got mutated. Because it got mutated, one of three things can happen. It will be only mildly affected. It will be a very trivial sort of mutation. The enzyme will be formed normally, but it will not be able to function as efficiently as the normal enzyme. So products are produced, but its quantity or the efficiency with which the products are produced is a little less. Secondly, what could happen is the product that is produced, the enzyme that is produced, its shape itself changes. If the enzyme shape changes, it will not be able to do its job. So the products are not formed. Worst case, the modification is such that the enzyme itself is not produced, in which case obviously the products will not form. So whenever such modifications happen, we can see newer phenotypes. Okay, so that is what dominance is. Usually the normal allele will be dominant and the modified one or the mutated one will be recessive. Now let's move on to another concept called codominance. This is also a deviation from Mendel's dominance. Mendel said, according to the law of dominance, whenever a pair of dissimilar alleles exist together, one will dominate, one will express, the other one will not. But in codominance, we will find that whenever a pair of dissimilar alleles exist, both of them can simultaneously express. There is no dominance or recessive. So we call that codominance. Co means together. This can be observed in the case of AB blood group. So you have different types of blood groups in humans. AB and O determined based on which antigen is present on the surface of RBCs. So in AB blood group, you have both A antigen and B antigen. Blood groups are controlled by a gene called I gene. I gene exists in three forms, three different alleles, IA, IB and lowercase I. IA allele, if it's present, it is responsible for the production of A antigen. IB allele, if it exists, it's responsible for the production of B antigen. Lowercase I will not produce either A antigen or B antigen. There is no antigen. So in A blood group, you will notice how only A antigens will be there on the surface. In B blood group, you will have RBCs having only B antigen. In AB blood group, you will have both antigens. And in O blood group, you have neither of those antigens. Okay. So what happens in AB blood group is, look at its phenotypes. You have four different phenotypes in blood group, O, A, B and AB. In O antigen, in O blood group, no antigens are there. So both the alleles will be lowercase I, lowercase I. In A antigen and B antigen, it can either be homozygous or heterozygous, but only at least one I capital A, I superscript A or I superscript B have to be there. Those alleles, at least one of them have to be there in the genotype. Only then it is possible, A blood group or B blood group is possible, that can exist either in homozygous condition or in heterozygous condition. When you come to AB blood group, you have to have one copy of IA allele and one copy of IB allele. So when both of them are present together, it's not like only one of them will express, as Mendel said, both of them will simultaneously express. That is why AB blood group is an example for codominance. So if you look at the different genotypes for A blood group, B blood group, AB blood group and O blood group, you will notice that only in AB blood group codominance is seen. In AB blood group, you have heterozygous condition. Even in heterozygous condition, both are simultaneously expressed. That is why it is codominance. But if you look at A blood group being heterozygous and B blood group being heterozygous, even though they are in heterozygous condition here, they are not showing codominance. They are showing complete dominance as suggested by Mendel. So when you're asked for an example for codominance, you can't write blood group. You have to write A blood group or B blood group. Be sure of, or AB blood group, be sure of it. So complete dominance is seen only in AB blood group. It is not seen in A blood group or in B blood group. There you can find complete dominance. Okay. Now, here are the various patterns of inheritance we've learnt about so far. In dominance, whenever dissimilar alleles are there, only one of them will express. In incomplete dominance, neither of them will fully express. You will find an intermediate phenotype, a brand new phenotype. In codominance, both the traits will simultaneously express. Okay, so that is called codominance. Now let's talk about multiple alleles. So for any gene, many different copies can exist in a population. Like if you look at the hair color of people, humans, there is individuals with black hair color, there are individuals with blonde hair, there are individuals with red hair. So many different traits for a character can exist. That exists because one gene can have many different types of alleles. If one gene has more than two alleles, then you call that multiple alleles. Though one gene can have more than two alleles, three or more alleles, one individual cannot have more than two alleles, because we are diploid individuals. In one individual, we have only two copies, two alleles present. But in a population, more than two alleles can exist for a gene. So that is called multiple alleles. ABO blood group is an example for that, because one gene I has three alleles, which is definitely more than two. So it is an example for multiple allelism. Next, we have concept of dominance. Uh, we cannot call a gene to be dominant, okay? Because sometimes one gene is responsible for more than one phenotype, for more than one trait. So we cannot say that dominance is an autonomous feature of a gene. I'll give you an example. There is one gene B, which exists in two forms, capital B, lowercase p. This gene B controls two traits or two characters, let's say. First is the size of the starch grains, and the second one is the shape of the seed. If it has both dominant alleles together, if both the dominant alleles are present, homozygous dominant stage, starch synthesis will happen very efficiently. So the size of the starch grains will be large and the shape of the seed will be round. If it is in heterozygous condition, size of the starch grains will be intermediate and the shape will be round only. If it is both homozygous recessive, then the size of the starch grains will be very small and the shape of the seed will be wrinkled. So if you look at this character, you are seeing incomplete dominance. If you look at this character, you're seeing complete dominance. The same gene is displaying complete dominance when it comes to the shape of the seed. Same gene is showing incomplete dominance when it comes to the size of the starch grains. So, hi Sanmukha. So, depending upon which character we are considering, we can't say that gene B is dominant, allele B is dominant, or allele lowercase b is recessive. We can't, recessive maybe you can say, but capital B is dominant, you can't always say, because with respect to this character, it is not showing complete dominance. Okay? A minute. So now let's talk about the dihybrid cross. Inheritance of two genes at a time. If you're considering two characters, then you call it a dihybrid cross. Mendel performed dihybrid crosses too, where at a time he selected two characters, both of them for the parental generation that he selected, both of these plants were showing contrasting traits. For example, if he considers two characters, let's say about the seed, one is the shape of the seed and the other is the color of the seed. One parent plant will show both dominant traits, the other parent plant will show both recessive traits. So when he performed his dihybrid cross experiments, he noticed that, okay, So he chose yellow and round seeded plants as one parent, green and wrinkled seeded plants as the other parent. This one had both dominant traits, this one had both recessive traits. In the F1 generation, obviously, both the dominant traits appeared. All of the plants produced seeds that were round and yellow. And when he selfed the F1 generation plants, in the F2 generation plants, he was surprised to see that newer combinations arose. Okay, so in the parental generation, you had only yellow along with round and green along with wrinkled. In the F2 generation, when he noticed the phenotypic ratio, 9 of them were round and yellow like this plant. 1 of them were green and wrinkled like this plant. But he had 3 plants that were round and green, 3 plants that were wrinkled and yellow. So he obtained 9:3:3:1 ratio where 9 of them were round and yellow like one parent, 1 of them were wrinkled and green like the other parent. But he got 6 different plants with newer combinations. 3 were yellow and wrinkled and 3 were round and green. He noticed that there were newer combinations. What did he take here? He took a plant that was yellow and round here, wrinkled and green here. In the F2 generation, F1 generation, he got yellow and round, all of them. But in the F2 generation, he got round and yellow, wrinkled and yellow, wrinkled and green. I'm sorry. Yeah, wrinkled and green, round and green. So here, I want you to notice how these combinations were already present in the parents. But yellow was never wrinkled before, green was never round before. He got newer combinations in the F2 generation. These are called recombinants. And the combinations that appeared from earlier, they were called parental types. Okay. So, yeah, based on his observations here, he proposed what is called the law of independent assortment. According to Mendel's law of independent assortment, whenever 2 pairs of traits are combined in a hybrid, the segregation of one pair of characters is independent of the other pair of characters. If the segregation of color was dependent on the segregation of the shape, then if they both were together, dependent on each other, then we would not have obtained newer combinations in the F2 generation at all. The reason we got newer combinations is because the color trait and the shape trait are not dependent on each other. They will segregate independently. That's why newer combinations were obtained. So that's about the Mendel's experiments. He published his work in the year 1865, but for as many as 30 plus years it was unrecognized up until 1900s. The reason is because communication was not very easy back in the day. He said that he said that factors are stable and discrete units that does not change and get passed on from one generation to another without any change, without undergoing any blending. But many people did not believe that because in nature you see continuous variation. If at all traits were passed on, factors were passed on from generation to next generation as it is without undergoing any change, then how do you explain so much continuous variation that you see in nature? That is one thing. Thirdly, he used mathematics to explain biological phenomena. Okay. But back in the day, many biologists did not understand that. That's one thing. And he said that within our cells, within our gametes, there are discrete units called factors, but he could not provide any physical proof for that. So these are some of the reasons why Mendel's work did not get recognized when it was published. Much later, in 1900, 3 people independently rediscovered his work. The 3 people are Hugo de Vries, Karl Correns, and von Schermack. So independently, they were working on different things and they chanced upon Mendel's work. And this happened in 1900 at around the same time. By the year 1902, microscopy was so advanced that people had I don't know about that, Prabha. Uh, Maybe not in this week, maybe in the next week you can expect. Okay? Yeah. So, around that time, by the year it was 1902, many more advancement had happened in the field of biology, and people had already worked out how chromosomes behave during meiotic cell division when gametes are forming. What exactly are chromosomes? What exactly do chromosomes do? They make an extra copy of themselves before cell division. They align at the equatorial plate. After meiosis 1, the homologous chromosomes separate. After meiosis 2, the sister chromatids separate. All of this had been worked out. They knew that crossing over and all of that will happen. Okay. So the entire movement of chromosomes during meiosis had been worked out by then. At around that time, two people, Sutton and Boveri, they kind of studied Mendel's work also because it was rediscovered in 1902. They also knew that chromosomes are behaving this way during meiosis. So they kind of combined both of these ideas. They knew from Mendel's work that Mendel said factors come in pairs. They also knew from chromosome work from what had happened after the movement of chromosomes were discovered. They knew that chromosomes also occur in pairs. From Mendel's conclusions, they know that factors segregate during gamete formation. That's the law of segregation. But they also knew that during gamete formation, chromosomes segregate. They had studied from Mendel's work that factors, when they segregate independent of each other. That is Mendel's law of independent assortment. They also knew that when chromosomes are segregating, they also segregate independent of each other. So Mendel could not provide physical proof for the uh, factors, but these guys united the knowledge of uh, chromosomal behavior along with Mendel's conclusions, and they kind of proposed what is called the chromosomal theory of inheritance, where they said the genes that Mendel was talking about are actually located on chromosomes. Because they are located on chromosomes, as and when the chromosomes segregate, the genes also or the factors also will get segregated. So they kind of gave us the physical location of the factors that Mendel could not. Now, in order to test whether the chromosomal theory of inheritance is true or not, uh, Thomas Hunt Morgan tried to experimentally verify it. He performed dihybrid crosses, this time using an animal, not a plant. He used the fruit fly for his experiment, which is called Drosophila melanogaster. Because of the same reasons that Mendel chose the pea plant, it had a very short lifespan. After each cycle of mating, it would produce many offspring. It had many variations. You could easily distinguish between the male and the female, and it was easy to grow in laboratory conditions. A simple synthetic medium could grow these insects very well. So these are some of the reasons why he chose the fruit fly for his experiment. So basically what he did was he conducted dihybrid crosses. He knew for sure whichever character he was considering for his dihybrid crosses, the genes responsible for those characters were located on the X chromosome. In Drosophila, similar to humans, males have one X and one Y chromosome and females have two X chromosomes. So males would have one copy of these alleles and females would have two of those copies. For one of his dihybrid cross experiments, he chose two characters. One is the body color and one is the eye color. Body color could be brown or yellow. Eye color would be white and red. So here, yellow body and white eyes are considered recessive types or mutant trait. Brown body and red eyes is considered dominant or wild type trait. To represent yellow body, he uses white color, white body, white eye, W, brown body, Y plus, red eyes, W plus. These are the alphabets that he uses to represent these traits. Hi Chaitanya, good evening. Now, he uses a female which has both recessive traits and crosses it with a male that has both dominant traits. Okay, so after he crosses them, these are the parents. After he crosses them, he obtains the F1 generation. He picks one F1 male and one F1 female and performs a cross between them for the F2 generation, and that is called intercrossing. So when he performs this cross, he will notice that in the F2 generation, the proportion of parental types that he gets was 98.7 type and the proportion of recombinants that he got was 1.3%. So here you can see that it is not according to the dihybrid cross phenotypic ratio that Mendel got. What did Mendel get? Mendel got parental type 62.5% and recombinants 37.5%. Similar dihybrid cross Morgan has performed using fruit fly, but here he got more parental types, as much as 98.7% parental types and only 1.3% recombinants. So there is this huge deviation that you see from Mendel's ratio compared to the dihybrid cross ratio that you got from Morgan's experiment. So what they did here was, he performed another experiment, another dihybrid cross experiment using another two genes. Okay. So when he performed the experiment using the other two characters, which is eye color and wing type, he ended up getting ratio similar to Mendel's dihybrid cross. He got 62.8%, which is not too much of a deviation, and 37.2%, again not too much of a deviation. And after he saw these results, he concluded that whenever genes are present on the same chromosome, they are physically linked, right? It is the same DNA strand that is present, it's the same DNA molecule that is present. So depending upon how closely they are located to each other, their linkage strength will vary. If two genes are located on a chromosome very close to each other, whenever crossing over is happening during meiosis, the chances of them getting crossed over together is very high. So you will not find too many recombinants whenever linkage is very strong. When can we say linkage is strong? Whenever the two genes are located very close to each other, we can say they are tightly linked. Whenever the linkage is very strong, you will notice that the in the in the F2 generation, most of them will be parental types. You will have very few recombinants. Whenever there is loose linkage, they are present on the same chromosome, they are linked, but the distance between them is much far. The probability of getting recombinants will be very high. Okay, so based on these results, you can notice that here they got more number of recombinants because the distance between the two genes is very high. Here you got fewer recombinants because the two genes were located very close to each other on the same chromosome. So based on this, he coined the two terms, linkage and recombination. Linkage refers to the physical association of genes present on the same chromosome. Recombination is the generation of recombinants, non-parental gene combinations. Whatever combinations you do not have in the parents, if you come across those combinations in the F2 generation, then you call it non-parental gene combinations or recombinants. Okay. Now, what does he infer from this? Some genes are tightly linked, some of them are loosely linked. The first dihybrid cross he saw, the genes were very close to each other, so he got very few recombinants, low recombination. Whenever they are loosely linked, present distantly from each other, you can find a higher recombination. Now, in Morgan's lab, there was his student working, his name was Alfred Sturtevant. What he did was, he calculated the recombination frequency by performing many different dihybrid crosses using two different gene combinations. And based on the recombination frequency, he kind of plotted the position of genes on a chromosome. If any dihybrid cross gave too many recombinants, then they probably are present distant from each other. If after any dihybrid cross you got fewer recombinants and more parental types, that kind of indicated that the two genes that he had considered for his dihybrid cross were located very close to each other. So based on this, he started, making genetic maps, gene maps. His name is Alfred Sturtevant. Okay. Yeah. Now let's talk about polygenic inheritance. So far we've learnt about how one gene controlled one character. In polygenic inheritance, we will learn about how one character can be controlled by more than two genes. Whenever more than two genes are controlling one character, you call it polygenic inheritance. Example for that includes height in humans and skin color in humans. When you look at height or skin color, you don't have two contrasting traits here. there is a gradient that you will come across. No person is like either completely black or completely white. You have so many shades of black, so many shades of brown, so many shades of pale. So it's like a gradient there. And even when it comes to height, it's not like people are either 3 feet tall or 6 feet tall. There is a gradient. There are people who are 3 feet 2 inches tall, 3 feet 5, maybe not. Okay, younger kids, younger people, or 5 feet 3 inches, 5 feet 10 inches, whatever that is. You have a gradient when it comes to height as well. So such type of inheritance is called a polygenic inheritance, and generally in polygenic inheritance, you can see an environmental influence as well. Like for example, you might be born having genes responsible for a pale skin color or a lighter complexion, but if you end up going to the sun and playing in the sun every day, then you will become darker, right? That is the influence of the environment. You might have genes that will allow you to grow very tall, but you're not having sufficient quantity of nutritious food. So without that nourishment, without that nutrition, even though you have the genes, you will not be able to grow very tall. So environmental influence will be here. Okay, so that is called polygenic inheritance. Let's understand this with the help of skin color. In humans, the skin color, let's assume, is controlled by 3 genes A, B and C. So if it's controlled by 3 genes, every individual will have 6 alleles. Considering we are diploid, every gene will have 2 alleles. So we have a total of 6 alleles. Depending upon whether those alleles are dominant or recessive, the skin color will vary. Okay. In its dominant state, it will produce melanin. So more dominant alleles means darker skin color. Fewer dominant alleles means lighter skin color. Okay. So assuming the 3 genes are A, B and C, if all of them are dominant, then it is the dark skin color. All of them are recessive, it is light. So this genotype will have the darkest skin color. This genotype will have the lighter skin color. Depending upon how many dominant alleles you have, the skin color will vary. For example, if you look at this phenotype having 3 dominant alleles, it could either be A, B. In this case also you have 3 dominant alleles. In this case also you have 3 dominant alleles, and in this case also you have 3 dominant alleles, right? Regardless of which gene is dominant or recessive, here the number of dominant alleles is what will matter. That is why it is also called quantitative inheritance. Okay, so all of these 3 different genotypes will have a similar complexion because they have the same number of dominant alleles. Now let's talk about pleiotropy. Before that, I'll just come 1 minute. All right. So we'll talk about pleiotropy now. In Mendelian genetics, we saw how one gene controlled one character. In polygenic inheritance, we saw how one character was controlled by more than 2 genes. Now in pleiotropy, we'll understand how one gene is responsible for more than one phenotype. Many phenotypes are controlled by just one gene, that is called pleiotropy. So in pleiotropy, a single gene can exhibit multiple phenotypic expressions. Okay, so what happens here is, we'll take phenylketonuria as an example. It is a metabolic disorder that some people have. So in such individuals, there is one gene responsible for the formation of an enzyme called phenylalanine hydroxylase. Okay, so that gene will get mutated. Because that gene is mutated, that enzyme will not be produced. Because that enzyme is not produced, the amino acid phenylalanine is not, just a minute. Yeah, phenylalanine is not converted to tyrosine, because of which many phenotypic effects can be seen. In individuals that have this mutated gene which cannot produce phenylalanine hydroxylase, you can find there is reduction in skin pigmentation, there is reduction in hair pigmentation, they are mentally retarded. These are three different phenotypes, right? Because one gene is mutated, that one gene is responsible for all of these phenotypic effects. That's why it is called pleiotropy. One gene responsible for one character we learnt in Mendelian genetics. For one character, more than two genes are responsible, we learnt in polygenic inheritance. If one gene itself is controlling many phenotypic expressions, we call that pleiotropy, and phenylketonuria is an example for that. What is the reason how one gene can control so many phenotypic effects? That one gene probably produces an enzyme which is involved in a metabolic pathway. Since that pathway is not happening, all of these phenotypic effects you can see in such individuals. Now let's talk about sex determination, starting with humans. In humans, sex determination happens by chromosomes, chromosomal sex determination. So humans have a total of 46 chromosomes, of which 44 chromosomes are autosomes, and the last pair is considered to be the sex chromosome. Females have 2 X chromosomes, males have 1 X and 1 Y chromosome. When 2 X chromosomes are present in females, in their normal diploid cells, when they undergo gametogenesis, when gametes form, when eggs form, eggs will receive only one of the 2 sex chromosomes. So all of the eggs will have 1 X chromosome. They'll be haploid. And when the males produce sperms, the sperms can either have X chromosome or Y chromosome. So depending upon whether the sperm that is going to fertilize the egg carries the X chromosome or Y chromosome, the gender of the child will be determined. So here in the parental generation, you have this guy having XY chromosome, woman having XX chromosome. In his sperms, either X chromosome or Y chromosome will be passed on. One of the two, 50% of the sperms will receive X, 50% will receive Y. 100% of her eggs will receive the X chromosome. If the egg containing X chromosome is fertilized by a sperm containing X chromosome, it will end up becoming a female. If the egg containing X chromosome is fertilized by a sperm containing Y chromosome, the offspring will end up having XY chromosome, which is found in males. So they will end up having a male child. Okay, so that is how sex determination happens in humans. Now moving on, let's talk about how sex determination happens in few other organisms. Before that, let me tell you about heterogamety. In this case, we saw that males produce 2 different types of gametes, females produce only one type of gamete. Because males produce 2 types of gametes, we call males heterogametic, females produce only one type of gamete, so female is called homogametic. There are 2 other examples where male heterogamety can be seen. Example in grasshoppers and also in Drosophila. In Drosophila, males have XY chromosome, females have XX chromosome. There you can find male heterogamety. In grasshoppers, this is a little different. The males don't have 2 different types of sex chromosomes. There's only one type of sex chromosome present. Males have one X chromosome only. Males have only one X chromosome. Females have 2 X chromosomes. So males will produce sperms either having X chromosome or lacking X chromosome. Depending upon which sperm will fertilize the egg, you can have either male or female. If the sperm does not contain the X chromosome and fertilizes the egg, it will be a male. If the sperm containing the X chromosome fertilizes the egg, it will be female. Now let's talk about female heterogamety. This can be seen in birds, where females will have 2 different types of sex chromosome and males will have one type of sex chromosome. Females will have ZW, males will have ZZ. Females, depending upon which chromosome they are carrying in the egg, whether they are carrying Z chromosome or W chromosome, the gender of the child will be determined. So if the female egg carries Z chromosome, it will end up becoming a male. If the female bird carries the egg containing W chromosome, it will end up becoming the female. Okay. Now, let's talk about how sex determination happens in honeybees. We call this haplodiploid condition. In honeybees, males and females have different ploidy. Females are diploid while males are haploid. Females being diploid have 32 chromosomes. Males being haploid have 16 chromosomes. Females undergo meiotic cell division to give rise to eggs that have 16 chromosomes. Males already being haploid can only undergo mitosis. They will undergo mitosis to produce sperms that have 16 chromosomes. Once in a while, the fertile queen bee, the female, will go on a mating flight, and after mating, she will collect sperms from the males and store it within a basket-like structure within her body, like a sac-like structure within her body. And depending upon whether she will fertilize her eggs with the sperms or not, males and females will form. If she chooses to fertilize her eggs with some of the sperms that she has collected, then after fertilization, those fertilized eggs will give rise to the female worker bees in the hive. If she needs more drones in the hive, more male bees in the hive, the egg that she has produced will be parthenogenetically developed into the males. Okay, so for the formation of males, uh, you know, um, for the formation of male drones, male parent is not involved. Directly from the egg itself, the male drones will form. There is no involvement of the sperm. So it is said that honeybee males do not have fathers. They cannot even have sons because for the formation of male drones, sperm is not essential. However, they can have grandfathers and grandsons. Okay? Yes, you may be able to identify it under a low power microscope, Harshit. Okay. Now, let's talk about mutations. You know how we all are different from each other because of the different combinations of alleles that we have. I told you how earlier for different alleles to form for the same gene, the changes in the nucleotides happen because of mutations, right? So we learn a little bit more about mutations. Mutations are basically changes in the uh, nucleotide sequence of DNA. Mutations can happen at the chromosome level or it can happen even at the DNA level. So if it happens at the DNA level, that means at the nucleotide level, the sequence of nucleotides will be changed. When I say it can happen at the chromosome level, I mean to say that sometimes, chromosomes break, like the arms of chromosomes can break, it can get deleted. One chromosome's arm can break and go and get attached to some other chromosome. All of that can happen. We call that chromosomal aberrations. Okay, or we can call that chromosomal mutations. So in this chapter, we will learn a lot about that. Now, before talking about how how DNA nucleotide sequence changes, we should understand that it cannot happen randomly like that. There are some agents that will cause mutations to take place. Whichever agents or factors cause mutation, we call those mutagens. Mutation, mutation generating factors or agents are called mutagens. Okay. Now, mutagens can be physical, chemical or biological. Physical mutagens include radiations like UV rays. They are non-ionizing radiations. They are also capable of causing mutations. And certain ionizing radiations like X-rays can also cause mutations, which is why people are advised not to get X-rays done very frequently because that can cause mutations in your body. Unless it's absolutely necessary, you should not get it done, right? So they are physical mutagens. Speaking of chemical mutagens, you may have heard how the cigarette smoke has so many chemicals that can cause lung cancer and oral cancer and all of that. So cancer is also happening because of mutation again. So they are chemical mutations. And biological mutations, there are certain viruses and other biotic agents which are capable of bringing about changes in the nucleotide sequence. They are called biological mutagens. Okay. Now, we have to talk about pedigree analysis. See, whenever you have to study about the inheritance of a particular trait, it could be a disorder, it could be any trait for that matter, you will have to understand how it has been passed on from one generation to another. It's not like every trait gets passed on using the same inheritance pattern. There are so many different types of inheritance patterns. So in order to identify, like let's say there are two individuals who decided to get married, but there is a genetic disorder that runs in their family. Okay, some individuals in their family have gotten a genetic disorder. They want to be sure that their offspring does not suffer from that particular genetic disorder. So they want to find out what are the chances that if they have a child, it will suffer from that disorder. So they will have to analyze the previous generations and everything and find out what pattern is being followed there. Is that particular disorder because of a defective autosomal gene? Is it present on the X chromosome? Is it present on the Y chromosome? How does it get passed on from one generation to another? Are this couple safe, or do they have to be worried about something? So all of that can be determined using a pedigree analysis. So in a pedigree analysis, we use a couple of symbols. Instead of writing big stories that my grandfather's brother had this and they had 3 children and she married somebody else. So instead of writing big stories like this, by making use of symbols, we represent the family tree. Who had the disorder, who did not have, and all of that by you can represent that using these pedigree symbols. In a pedigree analysis, we use a circle for female and square for male. If that male or female is affected, the circle is shaded. If they are not affected or if they do not have that particular trait, then you just have the outline. Okay, so that's the first thing. Secondly, if the sex is unspecified, you're not sure of whether it's a male or female, then you draw a rhombus or a diamond shape. Okay. Now, if you want to represent marriage or mating between two individuals, it will always be between a square and a circle, between a male and a female. So you draw a line. And if it's closely related individuals, if it's a consanguineous marriage, then you represent that by drawing 2 lines. If you want to represent the offspring of a particular couple, you will have to represent them in the order of their birth. So first, if they had a male child, first should be a square. If first they had a female child, then it should be a circle. So this couple here first had a daughter. So circle is first and square comes later. And here you can see this couple has only one child and that is a male and that male is infected. So that's how it's represented. Sometimes you end up having in your family 5 offspring, but none of them were affected. Instead of drawing 5 things, you can just represent it like that. Okay. So these are some of the symbols that are used in pedigree analysis. Now, let's talk about some genetic disorders. Genetic disorders can occur because of mutation in one gene, in which case we call it Mendelian disorder, or it can happen because there is a mutation at the chromosomal level. The number of chromosomes is more or less compared to the normal number of chromosomes. Then you call it chromosomal disorders. In this chapter, we have to learn about both. Now, when you're talking about Mendelian disorders, depending upon whether the gene that is responsible for that disorder is present on the autosome or the sex chromosome, you can classify that into autosomal disorders or sex-linked disorders. Depending upon whether it expresses itself in its dominant combination or recessive genotype, you can also further classify that into autosomal dominant or autosomal recessive, sex-linked dominant or sex-linked recessive. Okay. So, we'll talk about Mendelian disorders first and then come to chromosomal disorders. Among Mendelian disorders, you have first color blindness. Color blindness is an X-linked recessive disorder. People who suffer from color blindness, the kind of color blindness that we are talking about, there are many different kinds of color blindness, but the one that we are talking about right now is a red-green kind of color blindness where individuals will not have the ability to distinguish between red and green colors. This happens because there is a defect in the cones of their eyes. The reason we are able to perceive different colors is because of the cones that are present in our retina, right? So, because one gene is mutated in such individuals, the cones, either red or green, will not be functioning properly, which is why they cannot distinguish between red and green colors. Color blindness does not mean everything appears black and white for them. In red-green color blindness, what normally looks like this to us will look like this to them. Okay, so they can't distinguish between red and green. Both will appear kind of like different shades of yellow. Okay, so that is red-green color blindness. So the gene that is responsible for red-green color blindness is located on the X chromosome. Okay, so if you know, humans have difference in X chromosome in males and females. Females have 2 X chromosomes while males have only 1 X chromosome. So in this case, females have 3 combinations possibilities. Females can, let's say X is the normal allele, X with a superscript C is the defective allele that causes color blindness. In females, you can see either they can be normal or they can have 1 normal and 1 defective allele, or they can have both defective alleles. Unless they have both defective alleles, they will not be colorblind. These are perfectly normal. Even though they have 1 defective allele here, they have another normal allele which can make up for the absence of this allele's functioning, right? So this allele will produce normal rods and cones, because of which they will be able to see color normally. However, they are carrying a defective allele, which is why we call them a carrier. Okay. So these are the three possibilities of genotype when it comes to females. But when it comes to males, that's not the case. Since they have only one X chromosome, either they are normal or they are colorblind. There is no concept of carriers when it comes to males. Now, it is easier for males to develop color blindness because for males to become colorblind, even if the mother is just a carrier, there is 50% chance. But for a female to develop color blindness, 100% her father has to be colorblind and her mother at least has to be a carrier. Only in that case, female can become colorblind. Okay. So, because such cases where a colorblind man meets a carrier female and has children is very, very rare, you can see color blindness more in the male population than in the female population. 80% of males have color blindness while only 0.4% of females suffer from this kind of color blindness. That is simply because it is an X-linked recessive disorder. Not just color blindness, for all X-linked recessive disorders, the chances of females developing that disorder is rare compared to males. Males have only two possibilities. If they receive even one defective allele, they will end up being colorblind. But if a female receives only one defective allele, the other allele is normal, they will end up having normal vision. Okay, so that's the story of color blindness. Like I mentioned, it is a sex-linked recessive disorder. So here, this normal allele is the dominant one and this one is the recessive one. You know that recessive conditions express themselves only in homozygous condition. Only when it is in the recessive condition can you see color blindness. Okay. What are oncogenes? Oncogenes are modified proto-oncogenes. So we all have what is called proto-oncogenes. When those proto-oncogenes get mutated in certain regions, they transform into oncogenes and they will make our cells cancerous. Okay. It's no way related to what we're doing here. Yeah. You will learn about it in another chapter called Human Health and Diseases. Now let's talk about hemophilia. Hemophilia is a sex-linked recessive disorder again. In hemophilia, what happens is it's a blood-related disorder, clotting-related disorder. Whenever we fall down, we get hurt, we start bleeding, and the bleeding will stop because a blood clot will form, right? For clotting of blood, there are many different clotting factors required. Here, in these individuals that are suffering from hemophilia, blood will not clot at all because one clotting factor is not produced. That clotting factor is not produced because the gene that is supposed to produce that clotting factor is mutated. And that gene is located on the X chromosome. Okay, so in such individuals, because of the absence of clotting factor, even if there is a small wound that happens, very little bleeding also can, very small wounds also or cuts also can lead to unstoppable bleeding, and the individuals, if they are not attended to immediately, could even die. Okay. So this again, since it is an X-linked recessive disorder, it's more common in males than in females. You rarely ever find any females that have hemophilia. Many females can be carriers, but it's very rare to find a female that is hemophilic, because what happens during the embryonic stage itself is whenever two X chromosomes carrying the defective allele for that particular clotting factor comes together, the pregnancy itself will not continue. The embryological development itself will not happen, and the embryo will be aborted naturally itself. So that is why you do not see females that are hemophilic very commonly you don't see it. It's more common in males than in females, like all X-linked recessive disorders are. So here again, if a male has to be hemophilic, even if the mother is a carrier, 50% chances will be there. But for a female to be hemophilic, father has to be 100% hemophilic, mother has to be at least a carrier, only then the female child can be hemophilic, and that is very rare, like I said, because it will get aborted in the embryonic stages itself. Now, apparently in Queen Victoria's family, there were many carriers and many hemophilic people. That is why hemophilia is also called the royal disease. Since this is X-linked recessive, even over here you do not find any male carriers. Females can be carriers, but males can never be carriers. Males are either hemophilic or they are normal. There is no concept of carriers when it comes to hemophilia in males. Now, moving on, let's talk about phenylketonuria. Phenylketonuria is an inborn error of metabolism. This is an autosomal recessive trait. Autosomal recessive means the gene that is responsible for that particular trait is present on an autosome. Since there is no difference in autosomes in males and females, these autosomal recessive disorders can be found equally in both males and females. It's not like males have it more than females or females have it more, none of those things. You can find it nearly equally in both of the sexes. So talking about phenylketonuria, what happens here is there is one gene responsible for the production of an enzyme called phenylalanine hydroxylase. That gene gets mutated. Because that one gene is not going to get, because of that one enzyme that is not going to get produced, you can see that such individuals will have mental retardation. They will not, even physically develop properly. Pigmentation is affected in such individuals. Skin color is very pale, hair color is very pale. Okay. And also you will notice how in their body a lot of many different chemicals will get accumulated. Okay. So why that happens is because this enzyme plays an important role in the conversion of phenylalanine to tyrosine. Because this enzyme is not produced, phenylalanine will not get converted to tyrosine. Phenylalanine will get converted to chemicals called phenylpyruvic acid and some of its derivatives. Because of that, a lot of these phenylpyruvic acid and its derivatives gets accumulated in its body. It gets accumulated in the brain, and because the kidneys cannot reabsorb it in large quantities, it can be excreted through the urine as well. So that is why it is called phenylketonuria. Because it can accumulate inside the body within the brain, it can cause mental retardation too. Actually, tyrosine is very important in the production of melanin, which is the pigment responsible for our skin color and our hair color. If tyrosine itself is not getting produced because of the absence of the enzyme PAH, pigment also will not get produced, which is why they have reduced pigmentation in the skin and the hair. Okay, so that's about phenylketonuria. This is an autosomal recessive trait. In your syllabus, you have all recessive traits. You don't have any dominant traits explained in detail. However, they have given the example of myotonic dystrophy for autosomal dominant trait, okay, or autosomal dominant disorder. All right. Ashwant. Yes, I am trying, Ashwant. Now, let's talk about sickle cell anemia. Sickle cell anemia is an autosomal, recessive trait. So here what happens is, you know how hemoglobin is a molecule that is involved in the transport of respiratory gases like oxygen and carbon dioxide in our body. Oxygen to a large extent, carbon dioxide maybe to a small extent, but it is involved in transport of respiratory gases. Each hemoglobin molecule contains 2 alpha chains and 2 beta globin chains. What happens in sickle cell anemia is the gene responsible for the production of beta globin chain is mutated. It has undergone a substitution mutation such that at the 6th position of the beta globin chain, normally glutamic acid should be there, but instead of glutamic acid, because of the substitution mutation, valine will be inserted in the chain, okay, during protein synthesis. Because valine is present in the chain instead of glutamic acid, whenever there is low oxygen conditions that this individual will experience, at that time the hemoglobin molecules will attach to each other, polymerize, become rod-shaped rigid structures and change the shape of RBCs. RBCs are normally biconcave. That allows them to transport oxygen to our tissues very efficiently. If there is any change in the shape of RBCs, that can become problematic for us. Okay, so because the hemoglobin in low oxygen conditions, probably when you go to high altitude areas where there is not sufficient air pressure, or maybe you're exercising too much and not breathing properly, so your body is not getting sufficient oxygen. In such conditions, what happens is these hemoglobin molecules that would have normally dissolved in the plasma of RBCs, cytoplasm of RBCs, will form long rod-like structures or polymers and change the shape of RBCs from biconcave to sickle shape. Okay, so that is why it's called sickle cell anemia. So when this becomes sickle shaped, the problem is it will not be able to freely move inside the blood vessels because it is rigid. It is no longer able to squeeze through capillaries. So it can clog and make clots within the capillaries and prevent blood flow to major organs, which can be problematic. So that is about sickle cell anemia. Now here there are these genotypes possible. HbA is the normal allele, which is dominant. HbS is the defective allele, which is recessive. Since this is an autosomal recessive disorder, unless you have both copies of defective allele, sickle cell anemia will not be seen. Okay. So when you have both recessive alleles, you can see the disease. If they are both normal alleles, obviously it will be normal. If there is one normal allele and one defective allele, that individual is a carrier, but they will not show the symptoms of sickle cell anemia. Okay. Now let's move on to thalassemia. Thalassemia is also an autosomal linked recessive disorder similar to sickle cell anemia and phenylketonuria. So what happens here is, again, this is a blood related disorder. Here also there is anemia because RBCs are not able to transport oxygen sufficiently to the tissues. Hemoglobin is affected. But unlike what happened in sickle cell anemia, over here the quality of hemoglobin chain is not bad. It's just that the quantity is not great. Whichever genes are responsible to form beta globin or alpha globin chains of hemoglobin, they will be either mutated or deleted, because of which sufficient quantities of alpha globin or beta globin chains will not form. If not sufficient alpha or beta globin chains form, then ultimately the hemoglobin structure will not be formed properly. If the structure of hemoglobin is not proper, then it will not be able to do its job properly. Its job is to deliver oxygen to the tissues. Because hemoglobin cannot function properly, such individuals will suffer from anemia. Okay, so that is called thalassemia. Here there are two different types of thalassemia, alpha thalassemia and beta thalassemia. Alpha thalassemia is caused whenever there is a mutation or deletion in the genes responsible for production of alpha globin chain that is present on chromosome number 16. On chromosome number 16, you have two genes responsible for alpha globin chain synthesis, HbA1 and HbA2. If more number of alleles are deleted or mutated, more severe is the thalassemia condition. For beta thalassemia, there is one gene responsible for beta globin chain synthesis located on chromosome number 11, that is called HBB gene. So HBB gene will make sure, if it is mutated, then beta globin chain will not be produced in sufficient quantities. If it's not produced in sufficient quantities, then the ultimate hemoglobin structure will be different, and because that is different, it will not be able to deliver oxygen properly to the tissues, resulting in anemia. Okay. Now, both thalassemia and sickle cell anemia are anemia. Tissues are not getting sufficient oxygen because of defect in RBCs. The reason why RBC is defective in sickle cell anemia is because the beta globin chain's quality is not good. So sickle cell anemia is a qualitative disorder, whereas in thalassemia, the reason why RBCs do not have, the hemoglobin structure is ruined in thalassemia is because of not sufficient quantity of the globin chains being produced. That is why thalassemia is called qualitative disorder. Now let's talk about chromosomal disorders. Whenever you have difference in the number of chromosomes present in an individual, or number of sets of chromosomes present in an individual, you call that chromosomal disorders. So why do normally humans are supposed to have 46 chromosomes, but sometimes some humans will end up having 45 or 47, more or less than normal. Then you call that chromosomal aberrations. The reason that happens is because during meiosis, during gamete formation, chromosomes would not have segregated properly, and the gametes would have ended up having more than necessary number of chromosomes. And when such gametes are involved in sexual reproduction, when such gametes bring about fertilization, then such disorders will happen. So in your syllabus, there are 3 disorders that you must learn. First one is Down syndrome. Down syndrome, you have 47 chromosomes. Normally, every chromosome we will have 2 copies, but in Down syndrome, there are 3 copies of the 21st chromosome. That is why it is called trisomy 21. So, what are some of the symptoms that you can see in Down syndrome? So, such individuals will be short and have a round head. They will have a furrowed tongue. Their mouth will be partially open and they'll always be drooling. Their palm will be very broad and they'll have a characteristic palmar crease that runs horizontally on their palm. Their physical, psychomotor as well as mental development will be retarded. Of course, you would have seen individuals that look like this. Okay, across the globe, whoever has Down syndrome, they will have the same facial features. They will appear Chinese-like. That's why it's also called Mongoloid idiocy. They will have very small eyes and they will have like a flat face. Yeah. Moving on, let's talk about Klinefelter syndrome. Klinefelter syndrome can be seen only in males. Here, normal males have 44 autosomes and one X and one Y chromosome. Individuals that suffer from Klinefelter syndrome will have 44 autosomes normally, but they have one X chromosome, one Y chromosome. In addition to that, they have another X chromosome. So they have 3 sex chromosomes, 2 X and 1 Y. So because they have an additional X chromosome similar to females, they will show certain secondary sexual characters of females also, such as breast development, which is called gynecomastia. These Klinefelter's individuals, when they are born, they have normal masculine development itself, but however, they will show certain secondary sexual characters of females also, and such individuals are sterile. Okay, they will not be able to have children of their own. They have 2 X chromosomes plus 1 Y chromosomes. Unlike most normal individuals that have only 2 sex chromosomes, they have 3 sex chromosomes. So the total number of chromosomes in Klinefelter's individuals are 47. Okay. Speaking of Turner syndrome, this is something that you can find only in females. Normal females have 44 autosomes and 2 sex chromosomes in the form of 2 X chromosomes. But in Turner syndrome, the individuals will have normal autosomes 44, but instead of having 2 sex chromosomes as X chromosomes, they have only 1 X chromosome. So they will have 1 chromosome less than the normal number of chromosomes in normal individuals. So they will have a total of 45 chromosomes. Such individuals are not very different looking compared to normal females. They might be slightly shorter and sometimes you can find like a webbed neck, but not always. But they will not show any secondary sexual characters like development of breasts or widening of hips, and they are also sterile. Okay, they are sterile because their ovaries are not fully developed. They are rudimentary, they do not produce eggs, which is why they are sterile. Okay, with that we have finished principles of inheritance and variation. We'll move on to molecular basis of inheritance. Just give me a minute. I need to drink some water before I get back. Okay, back. Let's get started with the molecular basis of inheritance. So we just saw how, you know, characters get passed on from one generation to another. We understood the principles that govern inheritance in the previous chapter. What exactly happens at the molecular level is something that we will explore in the chapter molecular basis of inheritance. It's more like you're looking at, when we say genes are responsible for all of our characters, DNA is responsible for the color of our hair, for how tall we will grow and all of that. How exactly is DNA responsible for that? At the molecular level, what exactly is DNA doing to be responsible for our characters is what we will, I'm sorry, excuse me, my throat is hurting. Uh, yeah, is what we will be learning in this chapter. Okay, so let's get started. So this chapter's hero is the DNA, obviously, genetic material in most organisms except a few viruses. So what is DNA? DNA is a long chain of nucleotides. We call that a polynucleotide. So the individual units present in DNA is what we call a mononucleotide. So it is a long polymer of deoxyribonucleotides. The length of DNA in an organism or a species is fixed. Okay. So how much amount of DNA I have? Normally, all normal individuals will have the same amount of DNA. We measure DNA length not in centimeters, millimeters, nanometers. We measure it in terms of how many nucleotides are present in that segment of DNA, okay, or in that length of DNA. So here are some examples that you must remember. Bacteriophage 5x174 is a single-stranded DNA containing virus that has 5,386 nucleotides. Moving on, you have bacteriophage lambda, which is also a virus that has double-stranded DNA as a genetic material, and that has 48,502 base pairs. E. coli has 4.6 into 10 to the power of 6 base pairs. It is a bacterium, and our human DNA haploid content, one set of chromosomes, has 3.3 into 10 to the power of 9 base pairs present in it. Now, I told you that DNA is a long chain of nucleotides. Each individual unit is called a mononucleotide, and the entire chain is called a polynucleotide. In order to form the chain, one nucleotide has to get linked with another nucleotide by means of phosphodiester bonds. Okay, so this is the chemical bond that links nucleotides together to form a long chain. Yeah. Now, a nucleotide, each bead has 3 components. Okay, so one is a sugar, one is a nitrogenous base, and a phosphate. So the sugar here is a pentose sugar, meaning it is made up of 5 carbons. This is a central molecule. To this pentose sugar, a phosphate group and a nitrogenous base will be attached. That is one bead, that is one nucleotide. You have many such nucleotides joined to form a long chain of polynucleotide. DNA has 2 such polynucleotide chains. Okay. Now, let's talk about each of these individual components present in a nucleotide, starting with the sugar. The sugar, like I said, is called like I said, is a pentose sugar, meaning it has 5 carbons. This is the central molecule in the nucleotide. So here you can see in nucleic acids, you have 2 different types of sugar, ribose and deoxyribose. Ribose is found in RNA, deoxyribose is found in DNA. The difference between ribose and deoxyribose is at the second prime carbon. This pentose sugar has a ring-like structure where you have 5 carbons present. So you will start numbering the carbons from here in the clockwise direction. This is the first, second, third, fourth. 4 carbons are within the ring. 5th carbon is outside the ring. The same applies for deoxyribose too. Here you can notice how we are not numbering it 1, 2, 3 but 1 prime, 2 prime, 3 prime. The reason is because the nitrogenous bases also contain carbons. There shouldn't be any confusion while numbering them. In nitrogenous bases it's 1, 2, 3. In sugars it is 1 prime, 2 prime, 3 prime. Except for the 2 prime carbon, everywhere else there is no difference between ribose and deoxyribose. In 2 prime carbon of ribose you have 1 hydrogen and 1 hydroxyl group. Whereas in 2 prime carbon of deoxyribose you have 2 hydrogen. 1 oxygen less here, so it is called deoxyribose. At the second prime carbon, You have one oxygen less, that is why it's called 2' deoxyribose. Now, speaking of the second component, which is a nitrogenous bases contain nitrogen, carbon, hydrogen, oxygen, all of these. And they are of two types. You have purines and pyrimidines. Purines have a big name, small name, but a big structure. Purines have two ring structure. You can see both of them have double ring structure. And pyrimidines have a single ring structure. Big name, but small in structure. Okay. Purines are of two types, adenine and guanine. It is represented in short as A and G. Pyrimidines are of three types. You have cytosine represented as C, thymine as T, and uracil as U. Among this, adenine, guanine, cytosine are found both in RNA as well as DNA. Thymine is found only in DNA. Uracil is found only in RNA. So DNA will have ATGC, RNA will have AUGC. T is there only in DNA, not in RNA. U is there only in RNA, not in DNA. Now moving on, let's talk about how the sugar is attached to the nitrogenous base. The first prime carbon of the sugar is attached I'm sorry, excuse me. Attached to the nitrogenous base by means of a bond that is called a glycosidic bond. Okay. So depending upon which sugar is attached to which base, you have different names for nucleoside. Sugar plus base together is called a nucleoside. So depending upon which sugar it is and which base it is, we give it different names. If the sugar is ribose and the nitrogenous base is adenine, you call it adenosine. If it's guanine, you call it guanosine. If it's cytosine, you call it cytidine. It does not have T, it has U. If it is uracil, you call it uridine. So based on which sugar, which base, you give it different names. Likewise for DNA nucleosides, if the sugar is deoxyribose and the nitrogenous base is adenine, you call it deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Okay. Now moving on, the third component is the phosphate group. I'll show you how the phosphate group is attached to the sugar. It is attached at the 5' end. If this is the first carbon, fifth carbon will be outside the ring. To the fifth prime carbon, the phosphate group is attached by means of a bond called phosphoester bond. Now together, sugar, phosphate and base, everything together, you call that entire structure a nucleotide. This is the monomer unit that repeats in the long chain of DNA. Again, depending upon which sugar it is, which nitrogenous base it is, nucleotides have different names. If the sugar is ribose and you have adenine as a nitrogenous base, you call it adenylic acid, guanylic acid, cytidyllic acid and uridylic acid. And if the sugar is deoxyribose, then you call the nucleotides deoxyadenylic acid, deoxyguanylic acid, deoxycytidyllic acid. You do not have U here, you have T. So you call it deoxythymidyllic acid. Now, let's understand the difference between a nitrogenous base, a nucleoside and a nucleotide. Adenine is a nitrogenous base. Add sugar to it, it becomes a nucleoside adenosine. Add phosphate to it, it becomes adenylic acid, which is a nucleotide. The nucleotide minus a phosphate will become a nucleoside. Nucleoside minus the sugar will become the nitrogenous base. Moving on, let's understand how a long chain of nucleotide is formed. So here we have our first nucleotide with the 3 components. And here you have the second nucleotide with the 3 components. So here you have the sugar, the base, and this entirely is the phosphate group. Nucleotide number 1, nucleotide number 2. So phosphodiester bonds formation happens between these 2 nucleotides. In the phosphodiester bond formation, the 3' carbon of one nucleotide and the 5' carbon of the second nucleotide will be involved. To the 3' carbon, be it ribose or deoxyribose, you have a free hydroxyl group attached. So this hydroxyl group reacts with the hydroxyl group of the phosphate group. See here, to the 5th prime carbon you have a phosphate group attached, to the 3 prime carbon you have a hydroxyl group attached. So this hydroxyl group attached to the 3 prime carbon and this hydroxyl group attached to the phosphate of the 5th prime carbon will react. One molecule of water is eliminated and a bond formation happens between these two, and that is what we refer to as phosphodiester bond formation. So you can see here, so this is the, my god, this is very wrongly done. This image I will correct it in your notes, okay? So that is how between these two a phosphodiester bond will form. This image is incorrect. I will correct it in your notes, okay? I didn't check before taking it. So the phosphate P itself is missing from there. I'll correct and give it to you. So that's how a phosphodiester bond formation happens. Now you have another nucleotide 3 coming underneath the nucleotide 2. So here this 3 prime OHs, this 3 prime carbons OH, and the next nucleotides 5 prime phosphates OH will react to form another phosphodiester bond. Okay. Yes, Nutan, I teach in PW Kannada also. I teach in English also. Ishika is late, no problem. Your new NEET aspirant for NEET is 11th and 12th books is enough. Of course, it is enough. NCERT is more than enough. If you understand that very well and practice questions related to NCERT very well, that is more than sufficient. So let's understand the elucidation of structure of DNA. Today we know DNA as a ladder-like molecule that is twisted, but many people, did research for very long periods of time, and that's eventually when we figured out that this is the structure of DNA. So we learn about some of the contributions of scientists, starting with Frederick Mischer. Frederick Mischer is the person to have identified DNA inside the nucleus as an acidic substance, and he called it nuclein, and this was way back in the year 1869. Later, Morris Wilkins and Rosalind Franklin, they both worked in the same lab and they performed X-ray crystallography on DNA. They basically crystallized DNA molecule and passed X-ray through it and studied the diffraction pattern and found out that DNA had 2 strands, it was helically coiled and all of that. Much later, there was another scientist called Erwin Chargaff. So he said that in a double-stranded DNA molecule, the amount of purines is equal to the amount of pyrimidines. Okay, so how much ever A, G is there in DNA, the same amount of C plus T will be there. Purine is equal to pyrimidine. That is what Erwin Chargaff said. Later, 2 people, Watson and Crick, James Watson and Francis Crick, they kind of put together the contributions of Morris Wilkins, Rosalind Franklin and Chargaff, and they proposed what is called the double helical model of DNA, which is what we know today. So according to the double helical model of DNA proposed by Watson and Crick, as the name itself indicates, double helical means 2 strands are there. So DNA is made up of 2 polynucleotide chains. In these 2 polynucleotide chains, the backbone of the ladder, it's like a ladder that is twisted. The backbone is made up of alternating units of sugar and phosphate. So you have phosphate here, sugar here, phosphate here, sugar here. They will alternate. Even over here you have sugar, phosphate, sugar, phosphate. That will alternate. That forms the backbone. And the bases will project inside. Okay. So you can see the nitrogenous bases are forming the rungs of this ladder here. The bases are projecting inside. And the backbone is kind of made by sugar and phosphate. Right? Now, you can see there is opposite polarity of the 2 strands. The 2 strands are parallel to each other, but polarity is different. In this strand, if towards this side you have free phosphate group, that will be the 5 prime end. To this end, you have a free OH group at the 3 prime end. So this is the 3 prime end. On the opposite strand, you have free hydroxyl group here. So this is 3 prime end. You have free phosphate here. This is 5 prime end. Why is it anti-parallel? Why could it not have been parallel? What if both the 3' ends were present towards the same side here, 2 5' ends were there? Then the two bases would not have been able to face each other, right? If 3' end was, I'm sorry, 5' end was here, then phosphate group would be here and nitrogenous base would be here. The two bases in the opposite strands would not have faced each other, and hydrogen bonds would not have formed, base pairing would not have been possible. So that is why they are anti-parallel in nature. So the two bases, I mean the bases that face inwards, between them bond formation happens, hydrogen bond formation will happen. So it's not like any base can form pairs with hydrogen bonds with any nitrogenous base. There is specificity there, that is called complementary base pairing. A purine will always pair with a pyrimidine. A will pair with T by means of 2 hydrogen bonds. G will pair with C by means of 3 hydrogen bonds. So that is called complementary base pairing. That is fixed. Okay, it cannot happen randomly. Moving on, the chain is coiled, twisted, of course, in a right-handed fashion, which means if you hold your right hand like this, the thumb indicates the axis of the DNA and these fingers indicate the direction in which it is coiled. So that is how it is coiled in a right-handed fashion. One complete turn of the helix is called a pitch, and that measures around 3.4 nanometers in length. Within one pitch, you can find approximately 10 base pairs. So the distance between two consecutive base pairs will be 0.34 nanometers. All right, just give me a minute. Uh, okay. Okay. Now, over here in a double helical DNA, these base pairs will be stacked one on top of each other parallelly. That will confer stability for the DNA molecule as a structure. Okay. Now, in our cells, DNA does not exist as naked DNA. If you think about how long DNA in our cells is, if you take just the DNA from one cell, it will extend to as much as 2 meters. But our cells are very small, it measures in microns. Within that, there is a nucleus that is even smaller. So if our DNA has to fit in there, it can't exist in its very long naked form. So in order to fit it very well in an organized way inside the nucleus, it will undergo certain amount of coiling. Like the thread that we use for sewing at home, you don't use the thread as it is, right? In order to make it more compact, you will wind it around a cardboard roll. So it is more compact and more organized. The cells also do the same thing. So there are various levels at which DNA can be coiled or DNA can be packaged. So we learn about that. In us, DNA is mostly found inside the nucleus in interphase. So how packaging happens in eukaryotic cells we'll understand, and we'll also learn how prokaryotes package DNA. In prokaryotes, you don't have a nucleus. There is a region in the cytoplasm called the nucleoid where the DNA will be present. Okay. So DNA, because of its phosphate group, it is negatively charged, it is acidic in nature. So there will be certain positively charged proteins present in prokaryotes around which the DNA will wind. Proteins that are positively charged around which the DNA will wind, and it will form large loops that are held by few other proteins, and it will be present in a region called the nucleoid. What about eukaryotes? In eukaryotes, DNA is wound around in the first level of packaging. Packaging happens across many levels like this. This is naked DNA, it will get packaged like this, and further it will coil and become more and more condensed. So the initial levels of packaging, how it happens, I'm telling you. DNA is negatively charged in all organisms, eukaryotes or prokaryotes. Even in us, it is negatively charged because of the presence of phosphate group. So what happens is, in our cells, there are proteins called histone proteins, and histone proteins are made up of amino acids. Like all proteins, histones are also made up of amino acids, and the amino acids that are mostly found in histone proteins are arginine and lysine. Arginine and lysine in their side chains have a positive charge, because in the histone proteins, more number of arginine and lysine are there. It's not like the entire histone proteins are made up of just arginine and lysine. Other amino acids will be there, but the amount of arginine and lysine is more, and they carry a positive charge in their side chain, so that the net positive charge of the histone proteins will become positive. Okay, so they are basic and positively charged histone proteins. Our DNA is acidic and negatively charged. So there are 5 different types of histones present: H1, H2A, H2B, H3 and H4. So apart from H1, H2A, H2B, H3 and H4 in pairs, in multiples of 2, will form what is called a histone octamer. Around this histone octamer, the DNA will coil. Okay, so the negatively charged DNA is wrapped around the positively charged histone octamer. So it's like H2A, H2A, H2B, H2B, H3, H3, H4, H4. 4 of them in multiples of 2 will form the octamer. Around that, the DNA will coil, and H1 histone will seal it in place. Okay, so this is called a nucleosome. Okay, and when you look at such a structure under the microscope, see, like this is one histone octamer, there is coiling, H1 sealing, again coiling, H1 sealing. When you look at this structure under a microscope, it will appear like a string of beads, and we call that the chromatin. Okay. Thanks, Teju. So inside of our nuclei in eukaryotes, oh wait, further, okay, so this much I've told you. And further coiling for further condensation, from here for further condensation to ultimately form chromosomes, many other proteins are involved for the formation of chromosomes for further condensation and packaging. You know how packaging of DNA or condensation happens during prophase in mitosis, right? Or prophase of cell division. So during that, DNA will further coil, and for that, non-histone chromosomal proteins will be involved. We call them NHC proteins. So when you look at the nucleus, you can find 2 distinct regions inside the nucleus. One is lightly stained region, and the other one is darkly stained region. The lightly stained regions are called euchromatin, and the darkly stained regions are called heterochromatin. The lightly stained regions, I'll tell you about them. Why they are lightly stained is because the DNA is not completely tightly condensed there. It is loosely condensed, it's loosely coiled. It will take up less stain, that is why it is lightly stained. Over there, the genes are active, transcriptionally active. They are constantly transcribing and expressing themselves. Whereas in the darker regions, there is DNA that is more tightly coiled. It takes up more stain and appears darker. The DNA in those regions are not transcriptionally active. They're not expressing themselves at the moment. Okay, so that's the difference. So how did we know that DNA is a DNA is a genetic material? Many people performed many experiments to find out what is the genetic material's nature. Is it a carbohydrate, protein, lipid, RNA, DNA? What is it? Many people were trying to find out. So what happened was, one person called Frederick Griffith, he performed an experiment using mice and a bacterium called Streptococcus pneumoniae in the year 1928. Streptococcus pneumoniae bacteria were of 2 types. They were of 2 strains. One is called the R strain and one is called the S strain. S strain Streptococcus pneumonia bacteria, they had smooth colonies on their surface, they formed smooth colonies in the medium because they had a capsule around themselves. Okay, so they were encapsulated, which is why they were shiny in appearance. Whereas the R strain bacteria, they appeared rough in the colonies because their colonies appeared rough because they did not have a capsule. Okay, capsule provides that slimy, shiny nature. Since they did not have capsule, they appeared rough in colonies. Now, Streptococcus pneumoniae, as the name indicates, is known to cause pneumonia. It causes pneumonia in mice. This S strain that had the capsule could cause pneumonia, while the R strain that did not have the capsule lacked the ability to cause pneumonia. Since it causes pneumonia, it is called the virulent strain. This does not cause pneumonia, so it's called non-virulent strain. What they did was, he took the S strain bacteria, which is capable of causing disease, and injected that to mice. Mice obviously developed the disease and died. He injected R strain bacteria into mice. Mice lived happily ever after. Nothing happened. It was very happy. Then after that, what he did was, he took this live S strain and killed those bacteria. Okay, he heated them to a point where they died. He heat killed them and injected them into mice. Obviously, dead bacteria can't do much. So, no infection, mouse survived. So, with this, mouse survived, mouse survived, this mouse died, mouse survived, mouse survived. Then what he did was, he mixed these two. He mixed these two individually. When he added them to mice, mice had survived. He mixes them both and injects them into mice. When he injects that mixture, he expects the mice to survive. But to his surprise, mice will develop pneumonia and die. And when they performed an autopsy of the mice and look inside its body and checked for whether S strain bacteria were there, they will find this bacterium inside this mouse's body. But had they injected that in the first place, they hadn't. They had injected this one and this one, but they ended up finding that one. That means this bacteria must have transformed into this bacterium. How is that possible? So, the heat-killed S strain bacteria releases, there is some component of the heat-killed S strain bacteria. It could be its DNA, it could be its protein, it could be its RNA, whatever that is. Griffith is not sure of it. He says that there is some component of heat-killed S strain bacteria that is transforming live R strain bacteria into live S strain bacteria, and he calls that the transforming principle. So far, we don't yet know whether DNA is a genetic material. He just says that there is something in the heat-killed S strain bacteria that is making R strain into S strain. What did not have a capsule has ended up developing a capsule. So he calls that process transformation, and he calls that component as the transforming principle. Okay. Now, 3 other people will continue his experiment to find out what could be the transforming principle. Could it be RNA, DNA, protein, or what is the physical nature of it or the chemical nature of it? He wanted to find out. People wanted to find out. So 3 people, Avery, McLeod and McCarty, will continue his experiment. So the problem here was, he did not know which component of this transformed the bacteria, right? So this has carbohydrate, it has proteins, it has DNA, it has RNA, all of that. He's not sure of which one of these caused transformation. So what they first do is, they will take this heat-killed S strain bacteria and separate out its DNA, RNA, proteins and carbohydrates. Okay. So they will separate that. And to those separated components of the heat-killed S strain bacteria, they will add live R strain bacteria. And they will notice wherever DNA of heat-killed S strain bacteria was there, over there transformation will happen. In none of the other cultures, transformation is seen. So this kind of gave them proof that DNA itself is a genetic material, because wherever DNA was there, only their transformation happened. So in order to further confirm that, what they did was, they took the heat-killed S strain bacteria, separated them out into different cultures. To one they added DNA digesting enzyme DNase, to one they added RNA digesting enzyme RNase, and to the other they added protein digesting enzyme protease. Further, when they added R strain bacteria to that, transformation happened only where DNA was not affected. Oh, Bharat, namaskara. Good evening. Okay. So here they noticed whenever DNA was broken down by adding DNase, transformation did not happen. But whenever DNA was intact, then transformation happened. Okay, so this kind of indicated that for transformation, DNA is essential and gave them proof that DNA itself is a genetic material. But many people thought that DNA is never naked. It is never by itself. It's always associated with proteins. So it's possible that the proteins associated with DNA is what is responsible for transformation. And, you know, if not for, I mean, it's possible that the separation of these components of heat killed estain bacteria is not done properly. That is why probably they're getting these results. Many people did not buy it then. So this was not final proof for DNA as a genetic material. Ultimately, another experiment was performed by two people, Hershey and Chase, Alfred Hershey and Martha Chase. Hi Manisha, good evening. Alfred Hershey and Martha Chase. So what they did was they experimented with bacteria and bacteriophages. They are viruses that cause infection in bacteria. They took two types of bacteriophages, T2 bacteriophage, and they grew it on two different cultures. One contained radioactive phosphorus and the other one contained radioactive sulfur. Radioactive phosphorus is P32, radioactive sulfur is S35. Whichever bacteriophages were grown on radioactive medium containing P32, they incorporated that P32 into their DNA and became radioactive. I mean, they had radioactive DNA or radio labeled DNA. And the bacteriophage that were grown on a medium containing S35 incorporated those proteins into their capsids, okay, inside into the protein coat that they have. Okay. Check. I guess I'm audible now. Yeah. So whenever it was grown on a medium containing P32, the DNA was radio labeled because DNA has phosphate group, but the protein coat was not labeled because proteins do not have phosphorus. Okay. Now, what they did was the experiment in 3 steps. Firstly, they will infect 2 subsets of bacterial cultures with these 2 different types of bacteriophages. Okay. So that's the first step. They will infect. After that, they will do something called blending. Whenever blending happens, if at all any viral component does not enter the bacterial cell, that will be retained outside. It will be removed. Okay, we can't see what is happening here. We are doing these experiments and later we will come to this conclusion. Okay. So next step is called blending and the further final step is called centrifugation, where they will take this mixture and spin it in a centrifuge instrument at very high speed, as high as 75,000 to 1 lakh rotations per minute. When that happens, they will obtain in the centrifugation tube in whichever excuse me, in whichever bacteria had protein coat radio labeled, over there they will find radioactivity in the supernatant. In whichever bacteria DNA was radio labeled, they will find radioactivity in the retentate. Okay, in this pellet that is formed, they will find it. Okay, so here what you can say is, when you're performing centrifugation, separation happens based on density, right? So if you compare bacteria with viruses, bacteria are definitely more dense than viruses. So bacteria will tend to form pellets compared to viruses. So these bacteria did not by itself have radioactivity. But in this case, The bacteria are showing radioactivity. That means the radioactivity it obtained that property of radioactivity from the virus. In this case, which part of the virus was radioactively labeled? The DNA was radio labeled. So that means the DNA of the virus has entered the bacteria. That is why the bacteria is showing radioactivity, because the bacteria by itself was not radioactive. This kind of gives us proof that DNA itself is a genetic material. So Hershey and Chase's experiment provided unequivocal proof that DNA is a genetic material. So for a molecule to be called a genetic material, it should follow certain, it should fulfill certain criteria. Firstly, it should be able to make copies of itself. It should be able to replicate. Proteins, carbohydrates cannot replicate. DNA, RNA both can replicate. So both of them are genetic materials. Second, it should have stability, because that has information. Genetic material has information for pretty much everything inside the cell. It can't be changing very frequently. So it's important that whatever is considered as a genetic material, a molecule that is a genetic material, shouldn't undergo very frequent changes, and it should be stable, not just structurally but also chemically. Thirdly, it shouldn't be that the DNA or the genetic material does not undergo any change at all. It should allow scope for certain changes, because if changes does not happen in DNA, evolution will not happen. The reason we evolved from ape-like ancestors is because DNA changed and evolution happened. If the DNA remained as it is, evolution does not happen. So mutations, there should be room for mutation, but it should not happen very frequently. Very rarely, there should be scope for change, slow changes. Now, fourth point, it should be able to express itself in the form of Mendelian characters. So if you compare these properties with DNA and RNA, you will notice how both DNA and RNA are capable of undergoing replication and making copies of themselves. If you talk about chemical stability or stability in general, structural stability, DNA is more stable compared to RNA because of three reasons. RNA, it has a free hydroxyl group at the 2' carbon, so that is more reactive. Secondly, RNA has uracil instead of thymine, that is less stable compared to thymine. Thirdly, RNA has catalytic activity. It is involved in protein synthesis, formation of a peptide bond, and all of that. It will catalyze. So that makes it more reactive. But DNA is not like that. So DNA compared to RNA is more stable, providing scope for slow changes. Both RNA and DNA can mutate, but rate of mutation in RNA is much higher compared to the rate of mutation in DNA. And both of them can express themselves in the form of Mendelian characters. RNA can directly express itself, but DNA expresses itself via an RNA molecule. So that's why the reason why in majority of organisms DNA is a genetic material and not RNA is because of its stability. When the first living organism formed on our planet, uh, you know, um, it had RNA as a genetic material, because RNA has so many problems. It's so reactive, and it is less stable. It keeps changing very frequently. That is the reason RNA evolved to form DNA. DNA has evolved from RNA only. How do we know about this? There are many biological processes which have evolved around RNA. So that kind of gives us proof that RNA was the first genetic material, and because of its instability, DNA has evolved from it. And because DNA is so much more stable than RNA in most organisms that we see today, except for a few viruses, DNA itself is the genetic material. Okay. Now moving on, let's talk about how DNA expresses itself. When we say the color of my skin, DNA is responsible. How tall I'm going to grow, DNA controls that. The color of your hair, DNA controls that. The texture of your hair, DNA controls that, right? So we say that DNA controls every activity in our body. Pretty much everything that's happening inside of our cells is controlled by DNA. So how exactly does DNA do its job? DNA rarely ever gets out of the nucleus and does something, right? Unless the cell is dividing, DNA is always sitting inside the nucleus. Despite being inside the nucleus, how does it kind of control everything that's happening inside a cell? Just like you have head of institutions, like you have a principal or a dean in your schools and colleges, right? They make sure everything is functioning properly, but they themselves do not do all the work, right? They are the boss. They will have other people do work for them. So likewise, here DNA by itself does not do much. DNA makes the proteins do the job. So DNA is made up of nucleotides. Proteins are made up of amino acids. DNA can directly not form amino acids. Cannot produce proteins. So DNA produces proteins by means of an RNA intermediate, and different regions in DNA will have information for different proteins, and depending upon the requirement of the cell, those proteins will be produced and the proteins will end up doing the job. Okay. So when we say DNA is expressing itself, or let's say when we say skin color is determined by DNA, what exactly is happening? There is a DNA responsible for the formation of a pigment melanin in my skin. More melanin is there, we are darker. Lesser melanin, we are lighter in complexion, right? So, if we say that DNA is responsible for skin color, how is it responsible? There will be one segment in DNA. That segment of DNA produces a protein, let's say. That protein is an enzyme required for melanin production. So, if DNA produces that enzyme, melanin formation will happen. If DNA is not producing that enzyme, melanin formation will not happen. So, depending upon which gene you have there, what sequence of nucleotides you have, what allele you have, all of our phenotypic traits and pretty much everything inside the cell will be controlled. So, ultimately, it is the protein that does the job, but for the formation of protein, DNA has the master information. So, Francis Crick, who was one of the people who elucidated the structure of DNA, also proposed that DNA expresses itself unidirectionally from DNA to RNA and RNA to protein. That is how genetic information gets transferred unidirectionally. He says that DNA forms RNA by a process called transcription, and RNA uh, will undergo a process called translation to produce the protein. Okay. So DNA also makes a copy of itself by a process called replication. In this chapter, we're going to learn about replication, transcription, translation, all of that in detail. Okay. Now, okay. So we learn about replication. So many people gave many modes of replication, proposed many modes of replication. Some people said when replication happens, when DNA is making an extra copy of itself, the two existing DNA strands, I have no idea how long it will go on, Niharika. Maybe till 9 o'clock it will go on. The two existing DNA strands, both of them will be retained by one daughter cell and one brand new molecule will form. So many people gave different modes of replication, and some people suggested that of the two DNA molecules, of the two strands in the DNA molecule, one strand each will go to both the daughter cells, and both the daughter cells will receive one brand new strand, such that each daughter cell has one old strand and one new strand. This mode of replication was called semi-conservative mode of replication. Okay. So people were trying to prove how exactly replication takes place. So ultimately, two people, Messelson and Stahl, proved that replication happens semi-conservatively by performing an experiment using E. coli bacteria. So when they grew E. coli bacteria in a medium, in the medium, you have to provide bacteria with all the necessary nutrients, right? So they provided nitrogen in the form of ammonium chloride, and the ammonium chloride that they had used had a heavier isotope of nitrogen, not N14 normal nitrogen. It had N15. So these bacteria had N15 available. Whatever nitrogenous compounds that they are going to synthesize within themselves, they will utilize the same N15. For synthesizing DNA, they require nitrogenous bases, and for nitrogenous bases, they require nitrogen, and for that nitrogen, they are utilizing from the medium. And in the medium, what is available? N15 is only available. So they will incorporate that heavier isotope of nitrogen N15 within their cells, okay? And within their DNA also. How you can detect this is not by radioactivity. I'm not saying that nitrogen N15 is a radioactive isotope. It is a heavier isotope. So you can detect that using centrifugation. So they grew many bacteria, E. coli bacteria in a medium containing N15 for many generations, and they confirmed their result by performing centrifugation. Okay, so they perform something called cesium chloride density gradient centrifugation, where a density is already fixed. Depending upon where the band formation happens, you can say, okay, so this is the density, this is the density, you can figure out, okay. So obviously, if the DNA has heavier isotope of nitrogen, it will be more dense and it'll set down much bottom at the centrifugation tube, or if it's much lighter, it'll settle down much higher, okay. So after growing that in heavier isotope of nitrogen for multiple generations, when they spin it in a centrifuge, they obtain just one band corresponding to the density of DNA N15 and 15. We call this the heavy DNA. What they did after that was they transferred the bacteria into a medium containing normal N14 nitrogen. After every 20 minutes, they took samples and spun it in a centrifuge. Why 20 minutes? Because bacteria undergo cell division in every 20 minutes, and before it undergoes cell division, it has to make an extra copy of DNA. So DNA replication will happen once in every 20 minutes. So they took 20-minute intervals. After the first 20 minutes, when they spun it, they got a single band little above the previous band. Single band means every molecule of DNA there is similar. Okay. So there all of the molecules of DNA here were similar. They were all hybrids. If this is slightly higher than this, that means the density of this is slightly less than this. So obviously, right now they had transferred it into a newer medium containing N14. So they don't have N15, they have N14 here, which is why they will incorporate the N14 into the newly formed strands of DNA. So this hybrid DNA will be slightly less dense than this. In the next generation, all they have available is N14. So in the successive generations, you can notice that more and more lighter DNA will continue to get formed. After 40 minutes from the beginning after transfer, you will notice how there will be two bands that are formed. One band will be exactly as at the same position as this band, indicating that hybrid DNA is still present. Who is this? Okay, Sharanappa, hi. Good evening. And newer type of DNA is also forming, indicated by a new band. Okay. So this indicates that whatever DNA, other type of DNA is there, that is much lighter than this because it is settled much higher. So this kind of gave them proof that DNA replication happens semi-conservatively. If it happened conservatively, over here only they should have received two different types of bands. But here they did not get two different types of bands, which kind of proves that DNA replication happens semi-conservatively. So similar experiment was performed by Taylor and his colleagues using a plant called Vicia faba, which is also called faba beans. They used radioactive thymidine, and this also proved that DNA replication happens semi-conservatively. Now let's talk about how exactly replication happens. Replication in DNA begins at a particular region called the origin of replication. What we are learning right now is how replication happens in prokaryotes. Okay. If this is the double-stranded circular DNA you find in bacteria, there will be a specific region here called origin of replication, that is called the Ori site. Over there, there is an enzyme called helicase. That enzyme called helicase will break the hydrogen bonds existing between the two DNA strands and form what is called a replication bubble. What you're looking at here is one replication bubble. Each replication bubble will have 2 replication forks. I want you to notice how there are 2 fork-like structures here. This is one fork-like structure, this is one fork-like structure. Each replication bubble will have 2 such replication forks. We will take a look at what happens in one replication fork. The same thing will happen in the other replication fork as well. So, during replication, an enzyme called DNA dependent DNA polymerase will function based on the principle of complementarity. New strand will form. Okay. So here you have 2 strands of DNA, right? This is the parent DNA molecule that you're looking at here. These are the template strands that we have from the parent. Using these 2 template strands, newer strands have to form. So firstly, the 2 strands are separated. Each of these strands is used as a template. This is also template, this is also template. This DNA dependent DNA polymerase that I told you about, it is going to sit here. Okay, this, let me just call it DNA polymerase for now. So this DNA polymerase can add newer nucleotides at the 3' end. Okay, so at the 3' end, it will continue to add newer nucleotides. How it will add newer nucleotides is based on complementary base pairing. If over here this strand has A, here it will add T. If it has G here, it will add C. If it has C here, it will add G here. If it has G here, it will add C here. Like that. Based on complementary base pairing, it will add nucleotides. So when it adds nucleotides, it can work only from 5' to 3' direction, which means newer nucleotides can only be added at the 3' direction. It cannot add new nucleotides at the 5' direction. Because of this property of DNA polymerase, in both the strands continuous synthesis cannot happen. In whichever strand the template is 3' to 5' end, in that strand continuous synthesis happens from 5' to 3' end towards the replication fork. On the other strand, discontinuous synthesis will happen from 5' to 5' end, I'm sorry, 5' end to 3' end. Just because it is discontinuous does not mean it is any slower. It happens at the same rate, but on one it happens continuously, on one it happens discontinuously. This DNA polymerase is localized here. So to this strand it is easier to add nucleotides because 3' end is closer to DNA polymerase here. But here 3' end is far. So what it does is, it will take a little bit of DNA closer. There will be other proteins that assist in that. So it will bring the DNA template closer here, a small segment it will add. Again it will take another segment of DNA, small segment it will add. Like that it will continue. Okay. So yeah, sometimes they call this the leading strand, this the lagging strand, but that is something that it is not given in your NCERT textbooks. So wherever discontinuous synthesis happens, short segments of DNA will be formed. That is called Okazaki fragments. And to join the Okazaki fragments and make phosphodiester bonds between them, an enzyme called DNA ligase is involved. If DNA polymerase sees that A is here, T is here, G is here, C is here, in the template strand, it has to add, assuming this is the DNA polymerase, in the new strand, it must add T here, G here, C here, A here. I'm sorry. T here, C here, G here. Okay. So if this has to happen, then that requires energy, right? Nucleotides joining together to form a long chain requires energy. Anything big needs to be synthesized inside the cell, requires energy. So here what happens is, these nucleotides themselves will provide energy for the phosphodiester bond formation. So these nucleotides are, it will have deoxyribose sugar, it will have either one of the 4 nitrogenous bases, and at the 5' end it will have 3 phosphates. We call them deoxyribonucleoside triphosphates. It will have 3 phosphates. So if there is A here, there will be a nitrogenous base containing T here, it comes and binds to DNA polymerase, acts like the substrate here. Whatever energy is required for the bond formation, for the phosphodiester bond formation, that it will obtain by cutting these two phosphate bonds. Okay, so this will break the two phosphates. When that bond is broken, whatever energy is released, that is utilized for phosphodiester bond formation. So over here, these DNTPs not only act like substrates, but they also provide energy required for phosphodiester bond formation. Okay, so that is why we say that DNTPs or deoxyribonucleoside triphosphates will perform dual functions during DNA replication. Now, moving on. Yeah, another thing that I forgot to mention, DNA polymerase cannot start to synthesize DNA by itself. It requires short primers that are synthesized by an enzyme called primase that synthesizes short segments of RNA called RNA primer. Later, it will get replaced by DNA. So, that detail is not given in NCERT. Very briefly, in the end, somewhere they have mentioned, but I just want to let you know. Okay, so that's about DNA replication. Now, let's move on and talk about how transcription happens. Transcription is a process by which the information in DNA is copied by a strand of RNA. So this is something that happens inside the nucleus in eukaryotes. It happens within the cytoplasm itself in prokaryotes. So here again, the principle of complementarity is used. DNA does not get out of the nucleus. If it gets out of the nucleus, it can get mutated. So whatever information is there inside the DNA, if it has to get expressed, the message will get copied by RNA. It goes to the ribosomes for protein synthesis, and that is how expression happens. So the first step of gene expression is transcription, where the information in DNA first gets copied to a strand of RNA. Earlier in replication, we saw both the strands of DNA acted like templates. Over here, only one strand will act like a template, because right now we want RNA from DNA. RNA is single stranded. So for a single stranded RNA to form, you don't need two templates. One of the two strands of DNA will act like the template, and a strand of RNA will form. The process is called transcription. So this is also based on the principle of complementarity. The one difference is that in replication, if you saw A on a template, you would add T on the new strand. But here, if you see A in the template, you can't add T because RNA does not have T. You will have to add uracil containing nucleotide instead. Okay, so that's one thing. And secondly, in DNA replication, the entire length of DNA got copied, but here during transcription, only specific segments of DNA wherever information is there, and if this cell requires that information at that moment, only those will be transcribed. Okay, so why can't both of the DNA strands be transcribed? This is one question. Firstly, both of the DNA strands cannot be transcribed because if both of them are transcribed, then you get mRNA sequences of different, mRNA molecules of different sequences. The sequence of nucleotides in mRNA is what will determine the sequence of amino acids in protein. So if the sequence of nucleotides are different, two different nucleotide sequence in RNA you get from one DNA segment only, then we end up getting two different proteins from one segment of DNA, which will complicate the entire process of genetic information getting transferred, right? See, one segment of DNA is supposed to code for one thing. One segment of DNA is supposed to have information for one protein. But if both of them end up acting like template, you get two RNA molecules. Both RNA molecules have different sequence, because of which you get two different proteins. So it'll end up becoming like one DNA segment is responsible for two proteins. That will complicate the entire process, right? So that's one reason. Secondly, when the two DNA strands act like template, you end up getting two RNA molecules which are complementary with respect to each other. They will form double-stranded RNA, and double-stranded RNA will, will not be able to form a protein. It cannot be translated to form a protein. It will get broken down inside of our cells, and that's the reason why both the strands are not used for transcription. Now, I told you that there are only specific regions in DNA where transcription happens, specific segments only. So how would enzymes know where to start, where to end? So there is specific sites for that. The segment at which transcription is going to happen, the segment within which transcription is going to happen, that is called a transcription unit. A typical transcription unit contains three different components. You have a promoter, you have a terminator, and you have the structural gene. Promoter acts like the start site, terminator acts like the end site. Through the structural gene, transcription will happen. Whichever strand of DNA is involved in the acts like a template during transcription is called the template strand. The one that does not act like a template is called the coding strand. Then we have to designate the position of uh, promoter and terminator with respect to the code, with respect to the DNA strand, DNA molecule. We refer the coding strand. 5' end towards the coding strand upstream, you will have the promoter. 3' end from the coding strand downstream, you will have the terminator. Okay. Now, process of transcription involves an enzyme called DNA dependent RNA polymerase. This is the same principle of complementarity like replication. So what happens here is this DNA dependent RNA polymerase, it knows to read the nucleotide sequence on DNA and add complementary RNA nucleotides, but it does not know where to start and end. So in order to show it the start site, it will bind with a protein factor called sigma factor, and sigma factor will help identify the promoter sequence. We say that transcription is initiated at that point. After it has figured out the promoter region, it will break the hydrogen bonds in DNA itself and add complementary nitrogenous bases of RNA, and it will proceed till the terminator. This process is called elongation. RNA polymerase cannot determine whether the terminator sequence has arrived or not. In order to identify the terminator sequence, it associates with itself with another protein factor called the Rho factor. So once it associates with the Rho factor, it will figure out that the terminator end has been reached, transcription is complete, and everything will get dissociated. So that is how RNA will form. This is the story of how it happens in uh, prokaryotes. Okay, like in eukaryotes also it's similar only. But in eukaryotes, for different types of RNA being transcribed, you require different types of RNA polymerases. You have 3 different types of RNA polymerases in eukaryotes, RNA polymerase 1, 2 and 3. One transcribes 3 different types of rRNAs, which is 28S, 18S and 5.8S. RNA polymerase 2 transcribes a precursor of mRNA called hnRNA. RNA polymerase 3 transcribes 5S rRNA, snRNA and tRNA. Now, another difference in transcription in eukaryotes is that in eukaryotes, between the promoter and terminator within the structural gene, you have certain non-coding sequences called introns. They are intervening sequences that appear between expressing sequences called exons. Since they do not have information, before the RNA goes to the ribosome, you want to make sure that those unnecessary intron sequences are removed. Also, because you have to send the RNA through the nuclear pore to the ribosome for protein synthesis. But if you go to the cytoplasm, there are many nucleases that can act on the RNA and break it down. So in order to protect the ends of RNA from exonuclease activity, certain modifications will have to happen. So once RNA is produced, RNA will contain both introns and exons, and an RNA that contains both introns and exons is called hnRNA. Once this is formed, its 5' end is protected by adding a modified methyl guanosine, G-cap we call it. At the 5' end capping happens. At the 3' end polyadenylation happens. That means there is multiple A's, as many as 200 to 300 adenyl residues are added without a template at the 3' end. And by a process called splicing, the introns are removed and the exons are joined. And this gives us the functional mRNA. This is one other difference or one additional thing that happens in eukaryotes after transcription. So once these modifications are done, this functional mRNA is transported to the ribosome via the nuclear pore. It goes out to the cytoplasm and goes to the ribosome. So transcription is done. So let's talk about genetic code right now. You know that DNA produces proteins by means of RNA, right? So DNA getting copied to RNA is easy because both of them are made up of nucleotides. Complementarity principle is there, it's easy. But for RNA to form proteins, RNA is made up of nucleotides, but proteins are made up of amino acids. It's almost like those two are talking two different languages, right? So if the sequence of amino acids determine this, I'm sorry, if the sequence of nucleotides and RNA determine the sequence of amino acids in a protein, then there should be some code for which which nucleotide in RNA determines which amino acid. So if there is A, that means this amino acid should be there. If there is G, that means this amino acid should be there. Like that one code should be there. Unless that code is there, translation cannot happen properly, right? So people were trying to figure out what is the genetic code. Like if what nucleotide sequence is there in RNA, which amino acid will be specified. So very early on, one person called George Gamow suggested that there are a set of 3 consecutive nucleotides that will determine one amino acid. It's probable that the genetic code is triplet. What made him think so is because you have 20 different amino acids and you have only 4 different RNA nucleotides. If one nucleotide determined one amino acid, then you do not have sufficient codes for 16 amino acids. You have enough only for 4. And if it were a triplet, you have 16 combinations. Still, you are less of 4 codes, right? If it were a triplet, you will get 64 combinations, far higher than the number of 20 amino acids. So it's probably a triplet code is what George Gamow suggested. Later what happened was our Indian scientist Hargobind Khorana, he figured a way to synthesize RNA molecules with defined sequences, whichever sequence you want. That sequence of RNA you could synthesize in the lab. If you want like multiple use repeating, you can synthesize that using certain enzymes outside of cells in labs, in vitro, and whichever sequence you want. You want CU, 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 you can synthesize that. You want AU, 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 you can synthesize that. So whichever sequence you want, you could synthesize that. He figured that out. Then there was an enzyme called Severo-Ochoa enzyme which was used for this process. And then there was one person called Marshall Nirenberg who figured out a way to produce proteins without cells in cell-free systems. So, by making use of these people's contributions, finally they figured out that the genetic code is triplet. Hargobind Khorana synthesized these kind of nucleotides, chains of RNA of defined sequences. Using Marshall Nirenberg's cell-free protein synthesis, they figured that every time this was the RNA sequence, this was the amino acid sequence. So every time there are 3 U's consecutively, the amino acid that it is going to specify is phenylalanine. They tried this with other combinations of nucleotides, and eventually they even synthesized certain heteropolymers like this. Every time UCU was there, serine was the amino acid. CUC was there, leucine was the amino acid. So they did this repeatedly with different sequences and finally found out the genetic code. So this is the genetic code. You have a total of 64 codons that are specifying 20 amino acids. So the features of the genetic code is that the genetic code is triplet. You have a total of 64 codons, of which only 61 codons will code for amino acids. There are 3 codons that will not code for any amino acid. They are called the stop codons. They are UAA, UAG, UGA. There are certain amino acids that are coded for by more than one codon. Like in case of leucine, you have 6 codons. In case of arginine, you have 6 codons. Serine has 2. Proline has 4. Serine has 6 actually. Yeah, serine has 6. Arginine has 6. Alanine has 4. So one amino acid has more than one codon. That property is called degeneracy. And when amino acids are forming during protein synthesis, the mRNA molecule is read in a continuous fashion such that there are no spaces in between. No nucleotides in the RNA strand are skipped. So every nucleotide is a part of only one codon. It is read continuously. So that is the next property. Not necessary, Niharika. It's good if you can, however. And the next property is that it's nearly universal, which means if UUU in me codes for phenylalanine, and every organism pretty much is going to code for phenylalanine. We say it's nearly universal because it comes with exceptions. There are certain protozoan and mitochondrial genes where the codons are different. And ultimately, there is one codon called AUG that performs dual functions. It acts like the initiator codon, and it also codes for an amino acid called methionine. Okay. Now let's talk about mutations and the genetic code. The reason why mutations will have such drastic effects, as we saw in the case of sickle cell anemia, is because the sequence of DNA determines the sequence of RNA. The sequence of RNA determines the sequence of proteins. If in DNA there is any mistake in the sequence or any change in the sequence, then the sequence of amino acids also will change. Okay, so if the sequence of amino acids will change, the protein structure will change. If the protein structure changes, it will not be able to do its job well. It may not be able to do its job well. Okay, so that is the reason. I'll drink water and be back in just a minute. I'm feeling like extremely thirsty. Okay. So there are three different types of mutations that you can see. Point mutation meaning there will be change in only one nucleotide base pair. Okay. So if point mutations, you have three different types. You have substitution, insertion, and deletion. In substitution, there will be replacement of a base pair. For example, you can see in this case, in this codon, you have, where is the replacement? It's here. You have CTT here. This is the DNA, this is the RNA, this is the amino acid sequence. Since you had C on the template strand, you had G in the RNA. C got replaced with T. So that's why substitution mutation has happened. And because of this substitution mutation, instead of glutamic acid, lysine is added in the amino acid chain. So in the sequence of amino acids, there is clearly a difference. Next, you have another type of mutation called insertion. What happens in insertion is, within the existing nitrogenous base pairs, one base pair will be inserted in between. So here you had, excuse me, CTT. But yeah, in between this G and C, another C got inserted here, because of which from the point of insertion, every nucleotide, every codon has changed, and as a result, every amino acid also has changed. So we call this a frame shift mutation. Previously, after this change in base pair, there is no change in the other amino acids, only the ones where substitution has happened, only that codon is altered. But here it is not like that. From the point of mutation, every nucleotide base pair sequence or every codon will be changed, and every amino acid also will be changed. Next, you have deletion. In deletion, one base pair will get deleted. Here you had AAA, ATG. Now instead of ATG, because A got deleted from here, the reading frame has shifted. One nucleotide base pair from here will be moved to this. So it will become ACG, AAG, AG, next one. So from the point of mutation, every amino acid will be changed. Such mutations are called frame shift mutations. Okay. Now, if you have multiples of 3 nucleotides inserted or deleted, that will not cause a frame shift. In that cases, whichever codons are affected, only their amino acids will be altered, but after that, there won't be a frame shift mutation. Now, moving on, let's talk about translation. In translation, amino acid will join together to form a long chain of polypeptide of protein, and this happens inside the ribosomes. And for translation to happen, 3 different types of RNAs are required. mRNA is what is carrying the template for protein synthesis. tRNA is what will carry amino acids from the cytoplasm to the site of protein synthesis, which is the ribosome. And rRNA is a structural and catalytic component of the ribosome. It is within the ribosome that protein synthesis happens. Ribosomes are composed of rRNA. So rRNA is a structural as well as catalytic component. Why catalytic? Because for the formation of peptide bond, rRNA will catalyze the process. Okay. Now, let's talk about tRNA. tRNA acts like an adapter molecule. tRNA on one end will have an anticodon loop, which will have complementary bases to the codon present in mRNA. On the other hand, on the other end, it will have an amino acid acceptor end to which the corresponding amino acid will be attached. For stop codons, there are no tRNAs present. In its secondary structure, RNA will appear like a clover leaf. Like this, but in its final functional form, it will be like an inverted L shape. Okay. Let's talk about protein synthesis. Protein synthesis is when amino acids are joined together to form a long chain, a polypeptide. This happens inside the ribosome. The first step in protein synthesis is activation of these amino acids. First, these amino acids will react with ATP to form what is called amino acyl AMP, and a pyrophosphate molecule will get released. This amino acid attached to AMP is called activated amino acid. In the next step, the activated amino acid will bind to tRNA molecules. This process is called charging of tRNA. So once tRNA is bound to the amino acid, we say that the tRNA is charged. tRNA is not always bound to amino acid. When protein synthesis is happening, then it will bind to amino acid at its 3' end. Next, what happens? Initiation of translation. You have mRNA, functional mRNA 5' to 3' end. At the 5' end, you will have start codon. At the 3' end, you will have the stop codon, one of the 3 stop codons. To this mRNA, the small subunit of ribosome will bind, and to this small subunit, the first initiator tRNA carrying methionine amino acid will come and bind. To this, the large subunit will come and bind. Within the large subunit of the ribosome, you have 3 different sites. You have A site, P site, E site. The initiator tRNA will be attached at the P site. A site is called acceptor site. Depending upon which codon is present here, the next tRNA will enter from the acceptor site. So here you have UUU. UUU codon codes for phenylalanine. So this tRNA carrying phenylalanine will come and bind here. Once it comes and binds to the codon here, within the large subunit of ribosome, you will have a type of RNA called 23S rRNA that will catalyze the peptide bond formation between these two. Once peptide bond formation happens between methionine and phenylalanine, the tRNA which was attached to methionine at the P site will detach from it and exit the ribosome through the E site. It will get out of here. Next, what happens? Elongation should happen. This ribosome will slide on the mRNA, forming a long chain of amino acids. So when this slides over the mRNA, what was in the A site will come to the P site. A site will become vacant. So when it slides further, A site that was vacant will have another new tRNA come here, bind to it. Then Peptide bond formation will happen. This tRNA will get detached from this and move outside. Like this, it will continue to add amino acids and reach till the end of the mRNA molecule. Once it reaches the stop codon, you know that for stop codon, you do not have any tRNA molecules. Instead of a tRNA molecule, a release factor will come and bind here. Once release factor comes and binds here, all of the other components will get dissociated and the protein also will get, released. So this process is called translation. In translation, you have mRNA involved. Translation also proceeds in 5' to 3' direction. So in between the G cap and the start codon, you have certain regions that are not translated. Between the poly A tail and the stop codon, you have certain regions that are not translated. These are called untranslated regions or UTRs. Though they are not translated, they are important because they make sure that the translation process happens efficiently. Now let's talk about regulation of gene expression. Okay, see, you do not require all gene products at all times in a cell. Sometimes some products are not essential. So unnecessarily, if all the genes are getting transcribed all the time and translated, even if they are not essential, it's a wastage of energy because for during translation, during transcription, during replication, every time energy is getting utilized. So you should not waste energy for no reason. That's why the genes have to be regulated. Unless it is required, it should not get transcribed and translated. Okay, so that is regulation of gene expression. You can regulate the gene expression in prokaryotes at 4 levels. Firstly, you prevent transcription. If transcription itself does not happen, then proteins won't get synthesized. If you can't avoid transcription, I think I said prokaryotes, I mean eukaryotes. Second, once the RNA is formed, you prevent its post-transcriptional modifications. If that doesn't happen, translation will not happen. Even if that has happened, you can still prevent translation by preventing the transport of mRNA from the nucleus to the ribosome. Okay, so if that has also happened after transport at the translational level also, you can avoid the gene getting expressed. Okay, so at these levels you can regulate gene expression. In prokaryotes, it mostly happens at the transcriptional level. There is one example for this called lac-operon in your syllabus. So lac-operon, basically lac here means lactose. In E. coli bacteria, lactose metabolism is controlled by a set of genes that is called the lac-operon. Operon means it is a polycistronic structural gene that is regulated by a common promoter and regulatory genes. So we'll see what are the different components in a lac-operon. So here you have 3 structural genes, LAC-Z, LAC-Y, LAC-A. All of these 3 are regulated by a common promoter. You know how in a transcription unit you have a promoter and a terminator, right? So this is the promoter for these 3 genes. LAC-Z gene transcribes and translates to form an enzyme called beta-galactosidase that will break down lactose into glucose and galactose. Y will express to form an enzyme called permease that will increase the permeability of lactose into the bacterial cell. A produces an enzyme called trans-acetylase that will perform a trans-acetylation reaction which is involved in lactose metabolism. Now let's say there are these E. coli bacteria that are growing in a medium that does not contain lactose. If it does not contain lactose, then these enzymes are not required. You have to stop it. But let's say they are growing in a medium where lactose is present. If lactose has to be utilized, then these enzymes are absolutely essential. Usually if lactose is present along with glucose, glucose itself will get utilized, lactose will not be utilized. But if lactose is there and glucose is not there, the cells will prefer to use lactose and they will take it up inside the cell using permease enzyme. They will break it down using beta galactosidase enzyme and further metabolize it to obtain energy. Okay. So now that lactose is there, what will happen is there are certain regulatory sequences present here called I and O. Okay, so this is promoter for these genes. I already told you. I gene is a regulatory gene. It is called repressor gene. It will produce a protein called repressor protein. This repressor protein can bind to one of two things. It can either bind to operator here or it can bind to lactose. Okay. So if lactose is present in the medium, this will bind to lactose. If this binds to lactose, its shape will change such that it cannot bind to the operator. So what happens here? Our RNA polymerase will come and bind to the promoter along with the sigma factor. It will transcribe this, translate this, produce these enzymes and make sure to utilize lactose. But let's say lactose is no longer there. It is fully used up or the medium does not have lactose at all. If lactose is not there, what is the point of producing these enzymes? It's pointless. So there has to be some way in which the production of these enzymes should be stopped. So how do they do that? What happens then is this repressor that is getting produced continuously will have no option because lactose is not there, it cannot bind to lactose. The only option that it has is the operator. So it will go and bind to the operator. Once it binds to the operator, even if RNA polymerase comes and binds to the promoter, this operator physically is blocking the way. It cannot allow transcription of these structural genes. So transcription will not happen, translation also will not happen, enzymes are not produced. So these genes are switched off. Okay, so that is what lac operon is. Next, we have something called the Human Genome Project. It was a 13-year-old, 13-year-long project that was started in 1990 and it got completed in 2003. This was initiated by the USA. It was coordinated by two American organizations. One is US Department of Energy and the other one is National Institute of Health, NIH. So along with these two organizations, many other countries and organizations participated. So you have Wellcome Trust from UK, Japan, France, China and Germany. All of these countries also joined hands. The basic motive of Human Genome Project was to, sequence the entire length of DNA of one set of genetic information. Genome means one set of genetic information, haploid content. Okay. So in our haploid content of DNA, how much ever nucleotides are there, entire nucleotide sequence they wanted to determine. That is the Human Genome Project. But our DNA is so big, right? Even humans, one cells, one set of genetic information itself is so long. It has, if you were to store that in the form of books, I'll tell you about that. But first, let's talk about the cost. This Human Genome Project was called a mega project because it costed them a lot of money. In order to sequence one nucleotide base pair, back in the day, in the 1990s, it cost them $3. So we have 3 into 10 to the power of 9 base pairs in one set of genetic information. So if one base pair is costing $3, then this entire length of DNA would cost $9 billion approximately. Okay. So $9 billion is 900 crore dollars. Okay. So it was a very costly project back then. It was mega not just because of the cost. The amount of data that would get generated because of this was also huge. If you were to store that information in case of, in the form of physical books, assuming each book had 1,000 pages and each page had 1,000 letters, we would need 3,300 such books to store that much information. So it would have been impossible for us to store it in books, and analyzing it would become very difficult, which is why they try to store them and store them in computer programs. So let's talk about the goals of the Human Genome Project. Firstly, they wanted to determine the entire 3 billion chemical base pairs in DNA. Within the 3 billion chemical base pairs of DNA, 20 to 25,000 genes were present. They wanted to identify where those genes are. Whatever data they will generate, they wanted to store those information in databases. If they have to analyze the data, the existing tools for data analysis had to be improved. And if at all, whatever technology is developed for Human Genome Project, if it is useful in any other sector, they wanted to transfer related technologies to other industries. Because of this project, if any ethical, legal or social issues arises, they wanted to address that as well. Now, what are some of the methodologies that they used for Human Genome Project? Two methodologies. One is express sequence tags and the other one is sequence annotation. Expressed sequence tags, here no, only whichever genes are expressed, only those genes were sequenced. In sequence annotation, they took a blind approach of sequencing the entire genome regardless of whether information was there, they expressed or not, the entire genome was sequenced. How did they do that? They took one individual's, one cell's, one set of genetic information. They cut it into many pieces and they made many copies of that by introducing them into hosts like bacteria and yeast. So they cloned and increased the number of DNA copies. So they did this by using vectors called bacterial artificial chromosomes and yeast artificial chromosomes. Later, by using a sequencing technique called Sanger sequencing, they figured out using computer programs the sequence of DNA, and based on overlapping sequences, they finally produced the entire sequence of the entire length of DNA. So after the Human Genome Project, what did we get to know about the human genome? We got to know that a human genome contains 3,164.7 million base pairs. There are many genes that are there in our body, as many as 30,000 genes are present inside of our cells. So the length of these genes will vary, but on an average, every gene is, the average length of a gene is around 3,000 bases. The longest gene present in our cells is the dystrophin gene, which measures 2.4 million base pairs. In all individuals, 99.9% of the DNA sequence is the same. The difference is only in 0.1%. Of the 30,000 genes that we have discovered so far, we do not know the function of more than 50% genes. So the functions of more than 50% genes is not yet known. Among the entire length of DNA, the coding sequences are present in less than 2%. That is another thing we got to know from this Human Genome Project. Okay. I thought somebody is asking me if I'm sleeping in class. Move on. So within the DNA, there are many repetitive sequences. Okay. So certain segments of DNA are repeated hundreds to thousands of times. Chromosome 1 has the largest number of genes, which is 2,968. Chromosome Y has the least number of genes, which is 231. In the human genome, there are 1.4 million locations where there is difference between two different individuals in just one base pair. That is called SNP, single nucleotide polymorphism. We pronounce that as SNPs. So these are some of the salient features of our DNA. Apart from humans, many other organisms' DNA also has been sequenced. So bacteria, yeast, Caenorhabditis elegans, a nematode, Drosophila, Arabidopsis plant, rice plant, its genome also has been sequenced. So that's about that's about Human Genome Project. Now let's move on to DNA fingerprinting. DNA fingerprinting technique was developed by a person called Alec Jeffreys. If you want to identify the identity of a person, just like how our fingerprints are unique, our DNA sequences are also unique. Unless two individuals are monozygotic or identical twins, their DNA sequences will not be the same. So if you were to identify a person based on their DNA sequences, if you want to perform if you want to find out the entire sequence of their DNA, it will become very tedious. Too many people involved, too much money involved, too much time involved. So instead of having to sequence the entire length of DNA, we perform what is called DNA fingerprinting. Our total DNA length of one set of genetic information is 3 into 10 to the power of 9 base pairs. Between two individuals, 99.9% is the same. The difference lies only in 0.1%. So this 0.1% is also 3.3 into 10 to the power of 6 base pairs. If you were to sequence that much, that will also be too much time consuming and expensive for us. So what we do is, in our DNA, whatever repetitive sequences are there in the non-coding regions, we will utilize that and use that for identifying the individuals using DNA samples. So when you spin DNA in a centrifuge, you get two separate kinds of peaks. For bulk DNA, you get one big peak, and for our satellite DNA, which contains a lot of repetitive sequences, you get many small peaks. You isolate DNA from the small peaks and start with that. So within those, you have many satellite DNA. You get smaller peaks, no? That is called satellite DNA. You have many different types of satellite DNA based on the types of repeats you find there. Within satellite DNA, 100% you will find repeat. Check, check. Am I audible now? Check. The mic had run out of charge, so I am using a cord right now. I won't be able to go too close to the board. So I was saying that once the DNA is isolated, you will have to cut it down into small fragments. You do that using restriction endonucleases, after which you will separate the strands of DNA based on their size using gel electrophoresis. So once, depending upon the size, bands will be formed in gel electrophoresis. You will add an alkaline solution to it and make the double-stranded DNA single-stranded. Once that is done, you will blot the DNA that is present in the gel onto a membrane by a process called southern blotting. So when you do that, the DNA in your gel will be transferred to the nitrocellulose paper or a nylon membrane. Once that happens, you will have to do hybridization. So whatever is the VNTR that you have chosen, whoever is performing DNA fingerprinting, they will have an idea. In the lab, they would have artificially prepared radioactive probes complementary to your VNTR sequence. They will, incubate your nitrocellulose paper along with those radioactive probes. And if at all there is a complementary sequence, it will go and bind there. So after that binding process is called hybridization, where a single stranded DNA will bind with another radioactive probe and become double stranded. And then after that, this nitrocellulose paper is pressed against an X-ray film. Wherever probes are bound, there you can find bands in the X-ray film because of radioactivity. So that is your final DNA fingerprint. The kind of banding pattern that is the final DNA fingerprint, and that is unique for every individual. So with that, you will be able to find out the identity of an individual from a crime scene. Let's say you go to a crime scene and there are two suspects. There has been a murder and there are two suspects, and you want to find out, and you've got some sample from the crime scene. You want to know who was at the crime scene. So you take the blood sample of these two suspects and you compare that DNA fingerprint with the sample that you obtained from the crime scene. Whichever one matches, you can maybe take them for questioning and ask, how is it that your hair strand was there? Yeah, so that's what it is. So this DNA fingerprinting has more, like, a lot of applications, of which two are very important. One is it is used for in forensics, in like to find out the criminal and stuff, and it's also used for paternity testing. For paternity testing, 50% of the band should match. That will indicate that who is the father of the child. Okay. So yeah, I guess with that we are done. It's over for today. I'll see you again in the next class where we will be discussing. All right. I'll see you again. Thank you all for joining. Good night. Take care. Study well. Bye bye.